Okay, so we're going to start our first day. Welcome everyone to the Grapedia Annual Meeting. Hopefully not the only meeting that we will have as a collective, as an initiative. I welcome you through these 3 days. I hope that you enjoy the talks and the showcase of our prototype that we will do. Hopefully for next year, we will have it implemented. And I would like to also to thank Olga Mayoral from the Botanical Gardens, who allowed us to be here in this magnificent place, surrounded of amazing species. And I will give her the time to talk about the gardens. So please, Olga, thank.
Thank you, Thomas. Do you think you can put my source colors? Okay. Okay, while they're putting the PowerPoint, I'd like to welcome everybody to the Botanical Garden of the University of Valencia. In my name and the name of the director, Jaime Güemes, who couldn't be here. And I see that there are some colleagues, some researchers from the Botanical Garden there. So I'm really happy that this meeting is also interesting for our researchers. I'm going to spend just 10 minutes trying to explain to you some important facts about the garden so that when you go out in the coffee breaks and so on, you can enjoy it in a complete way. And I hope all the people who are online will be willing to visit us after my super short explanation of the garden. I think the ideal situation would be just to have a visit all together, walking around, but I'm sure you'll be able of doing it by yourself. So as Thomas said, I'm Olga Mayoral. I'm the deputy director of the garden, and I'm a botanist, but I'm also a teacher of didactics of science. So I'm teaching future science teachers, and you'll see this in my explanation because I'll tell you some of the things we're doing here in the garden. You have to pass it for me in front. Okay, so this is how it looks like. I don't know if you've had the opportunity of going around for a while, but it is an important green area of the city. We've got a very big green area down in the old riverbed side that connects many different places of the city, but this is one of the biggest green areas that is at the same level as the rest of the city. I'm going to try and pass it once more by myself, and if I can. Okay, yeah, I think it works. So, yeah, I think it's working now. This is how it looks like from the air. It's a garden of between 4 and 5 hectares, and it is completely surrounded by the city nowadays, but it wasn't that way in the past. The University of Valencia, that is more than 500 years old, has nearly from the beginning had a botanical garden, but it's changed of place. And so the origin of our botanical garden is similar to others in Europe. They began in the 16th century, and they were devoted to medicinal plants most of all, and they were linked to medicine studies. This has been changing along the history. And one of the important places where it was planned to be was in the Alameda. I say this because there are some people from Valencia who may know this place. Also really near to the old river. But nowadays it's here in what we call the Hort de Tramolleres. The university bought this orchard that was out from the city. You will see that there are the towers of Quad there besides here, but we are out from the old city. So this was a very far away place from the university. You can see on the right side a little map of the garden, and you'll see that there's the biggest part is blue. That's because that's the School of Botany. So the botanical garden was made to study plants. only the students that were studying plants came into the garden. This has changed a lot because nowadays the garden belongs to the city, although it's from the university, but people in Valencia think it's the garden of the city, and we're happy about it. But we've lost a little bit this connection with the studies of botany. We just visited some days, probably because the campus are quite far away. So the School of Botany was like the center, and there are other collections, about 20 and so on. And one of the important collections is the one you're devoted to, and I hope you will be able of seeing it. I'm going to try and show you the place so that you can search for the, okay. Here, okay, we are here. When you go through the principal avenue here, there's some magnolias around here, and there's this yellow building, which is the Orangerie, La Estufa Freda in Valencia. And just here, this purple part is the part you're most interested in. So I hope you will, it's quite easy. You do just have to go straight on and you'll find it. So there are about 20 collections, and each visit you make to the garden can be different. If you're interested in medicinal plants, in carnivorous plants, in endemic species, in adaptations, you can design your own visit. So I hope you'll do it that way. So the garden has been here since 1802. Most of the big trees that you'll be seeing are around 200 years old, okay? The garden has suffered different crises, and some of the architectonical elements were spoiled a little bit, but we maintain the original greenhouses, and you will be able of seeing them. And we also keep this is the shade house, which is one of the most important places in the garden, which is besides the greenhouses. Okay, you'll see it easily. This is how it was before the 80s, and the university really spent a lot of money in the 90s. And in the end, in 2000, this building was established, and all the researchers came here, and a good part of the garden was restored. And this is how it looks like now, the shade house. It is a very important element in Mediterranean climate areas where we can really make an environment that is bearable for species who need this shade. An important collection is the collection of palm trees. It is all spread out around the garden. You will also be able to see many species of palm trees that in other botanical gardens in Europe need greenhouses, but here they're completely outside. And there's an important, there are two important areas with native species and with some endemic species. The Valencian community, we've got more than 70 taxa that are endemic, they're only growing in the Valencian community, and we've got them there alive. But here, you won't have the time to visit the research building. We've got the seed bank, the germplasm bank, with all the species of plants of the Valencian community. We're not focused on ornamental or on species that are important for agriculture, but we also have in the seed bank some representations. Here you can see on the left side Camairops humilis, which is one of the only native palm trees in Europe. And then we've got other species. This is not the best moment to see them in flower, but you will see some endemic species around. So I'm sure you know this, but we are in the Mediterranean, and it is one of the Mediterranean basin with other areas of the world with a Mediterranean climate, which are areas that are really good for olive trees, for vineyards, for almond trees. These are places with a really high quantity of biodiversity, mostly of plants. So we've got really a responsibility in keeping this biodiversity, and that is one of the goals of this botanical garden. This is not only a collection of living plants, but it is also an ex situ conservation site. So we've got this seed bank or germplasm bank but we've got also an herbarium where we keep samples of all the species. We've got some molecular labs. We're making many research on seeds, and we really like to establish links with other researchers that are working with plants. So we're really happy to have you here, and we hope we can connect somehow. We also have many ornamental species, and we've got some invasive species like Cortaderia seyoana. You can see it on the left side. So we also talk about the importance of being really careful with invasions and with the globalization of the flora around the world. So there are many things that we can explain through the species we've got in the garden. Of course, you'll be seeing many signals. I want to tell you there are also some monographies speaking about some groups of plants or about the botanical garden. They're quite small. You can take them in the plane. The titles are in Spanish, but we've got them also in Valencian and in English. And well, I want to tell you a little bit about the research building, what we do here in this building. We're linked to the Department of Botany from our university. And I told you we work a lot with seeds, with spores of ferns also. And there's some iconic species we've been working with. And one of them is the one you see beneath. It's Cistus heterophilus subspecies cartaginensis, which is the most endangered plant in the Iberian Peninsula. These are some of the experimentations we're carrying out. We're seeing possible problems with reproduction. We are seeing which species of insects are helping to pollinate. We are seeing if there are problems of endogamy. And well, there's only one individual alive around here that's living in nature, but together with the local the regional administration, we are making some other new populations just in case. We've got many fires here, and we have to make sure we've got populations just in case. So this is how we work normally when we go into the country. And we work a lot also with ferns, and the group of people who came here today have been working a lot with ferns and with species that grow also in rockeries and cliffs, which is very difficult to try and germinate seeds and take them back to the cliffs. It's really, really difficult. Also, with species that grow in water. And these are all different groups of plants we're working with. And of course, everything we work with has to have a sample in the herbarium. The herbarium, we could think that it's a very old-fashioned way of working with plants, but it is really useful because it's having a sample of each species from which you can even get molecular information. You can also get some seeds in some occasions. We can get data from the phenology. We can get data if we've got samples from the 80s, from the 50s with flowers. We can see at what altitude certain species were flowering in this moment of the year 50 years ago, and we can make a comparison and see if this is evolving, if species are now flowering before. So there are many, many different studies we can do, and all these go to the GBIF database, the Global Biodiversity Database, where all the information about biodiversity goes, and it's available and free for everybody. So one of the important factors that we carry out here is genetic characterization of different taxa. And then I also wanted to talk a little bit, because I've been talking about preservation and also about research. But like the other important pillar of the botanical garden is education. And we are really worried about plant blindness, this phenomenon by which many people tend to not understand the world of plants. And useful plants, like the ones you're working with, are very, very they're like treasures, and we can use them to try and convince people of the importance of plants. Many people tend to, when you talk about living beings, they always think on animals, and when they try to approach plants, they see them as animal beings, and it's difficult to see them as attractive living beings. So that is one of our worries. And that's why we work a lot with students from different academic levels, trying to approach the world of plants to them. And we link it a lot with sustainability, because we are convinced that photosynthesis and the preservation of plant biodiversity is essential in this climate crisis we're living. And a good way of explaining it is through the plants that we've got here. We work a lot about nature-based solutions, and we work with future teachers. So we work in initial teacher training and ongoing teacher training of all levels. In this sense, we focus a lot on the importance of photosynthesis and how photosynthetic organisms are one of the clues that is helping us to reduce the quantity of CO2 in the atmosphere. So we try to focus on photosynthesis that we have all studied. We know by heart the formula, but do we really understand when we ask people where does the mass, so the material from plants, come from? They always tend to answer it comes from the soil. They never think it's a gas. It's the carbon from the atmosphere that is transformed into matter. So all these things are really important to explain, and it's very important to explain the importance of plants in the cities to try and make cities nice to be living in. In Valencia, you know, we've got really high temperatures, but the green areas, our temperatures are like refuges where you can go when it's really hot. So all these things can be explained easily in the garden. Just so that you know, you'll be staying here for some days also, only, but if you go to our website, you will see that there are many cultural things happening here. We've got 2 exhibitions now that you can see, one in the Orangerie and one in the Sala Tramoyeres at the entrance of the garden through the normal entrance. But we've also got concerts, we've got excursions with botanists who explain things about plants, and anybody can join these proposals. We've got you're not going to be here while we've got concerts, but we've got concerts with even with beer, and we've got some seminars, and we always try to bring conferences that have to do, or meetings with plants. We've also got this online publication that is very this dissemination publication, and we want to invite you, if you want to write things that all the citizenship can understand, you're welcome to do it in our publication Spores. And well, these are different moments of the garden. Sometimes we open in the night. We've got poems, we've got music, we've got people coming to tell stories. We've got scientists coming to explain their research. So there are many different opportunities of linking our research with the neighborhood and with the citizens. And of course, we've got many schools. You will be seeing them these days. You'll see groups of children around. I think with this, I hope you had a general idea of the garden, and then I invite you to go around, and welcome. Thanks for coming.
Thanks very much, Olga. I'm pretty sure that the whole audience will be very interested in go and visit the infrastructure and the plants that are in the garden. Definitely they should go and see the grapevine collection. There's very nice local grapevines there as well. And thanks for explaining the vision and the mission of the botanical gardens.
Yes, you're welcome. I hope you enjoy.
Okay, so again, welcome everybody, and thanks for joining this meeting. we're going to do a short welcome speech and thanking all the players that have been helping us since we started this project almost 1 year ago. So first, the project Grapedia was funded by the Cost Innovators Grant, also known as the CIG. And despite the CIG is coming to an end this year, we really hope that is one of the first of many meetings. So we will discuss this through these 3 days. I hope that you agree in the fact that we will be able to continue the meetings, because the topic that we will be talking about all around these 3 days is actually something that will it's growing, especially in the Grapevine community, and that we are continuously working on, which is computational biology, databases, and data. We are celebrating data, basically. First, I want to thank the organizing committee that has been helping me since we decided to do the annual meeting here. It has been very stressful for them, I guess. So big thanks to Aureliano and the 3 guys down there that I call the 3 musketeers of the Tom's Bio Lab. And I would like to comment that there have been fundamental, not only in the organizing this meeting, but also in the different activities that we have been doing during the year. So expected to be functional in 2021 as a beta testing model. The Grapedia portal is proposed as an open access platform. I think that you have learned about this during the whole year. You have listened to us on videos, on training schools and short-term scientific missions about our whole aim of the Grapedia portal. Our idea is to address challenges related to the accession of genetic, omics, and phenotyping data related to grapevines. Now, together with the showcase of a prototype, in this meeting, we have also gathered different talks that I hope that you will be able to enjoy, that are all around computational biology, that include data integration, pangenomes, annotation pipelines, a future, hopefully, promising QTL browser, and many, many things related to grapevine research. We hope you enjoyed these 3 days and explore the various facets of the Grapedia creation. I would like to acknowledge, and I hope that you have a little bit of patience for all the thanks that I need to give, but I would like to acknowledge all the players that have helped along the way for us to be presenting our prototype here today. So first, to the Grapevine community. Grapedia integrates all the resources generated by this community, fostering a comprehensive global grape information database. In other words, this portal is for you, by you. Since the first grape genome sequence was released, the grape community all over the world has generated an enormous amount of data. And this data is the data that we are collecting and offering it in a single platform. We hope that we are now ready to integrate all this data to enable researchers and the industry to design the vineyard of the future with more quality products, more resistant to pests, and climate transition. I would also like to thank the Cooperation and Science Technology Office. Not only they have funded us through the Cost Innovators Grant, the CIG, but also the Cost Action Integrate, from which Grapedia stems. Their funding has been essential to bring together this community. Their fundamental networking tools, all the ones that you see here, include meetings, training schools, scientific missions, and dissemination tools. We have taken advantage of all of these activities and all of these methods, and our team has taken full advantage of them to arrive to where we are today. All throughout this year as well, their assistance with coaching sessions has been fundamental. They have helped us in reaching the community in the best way possible. We have 2 very successful training schools, one in Barcelona and a second one in Trento, and the family has been growing since then, as you can see here. Cost has guided us in our communication skills workshop in how to also share data efficiently, in how to write a business plan, something so important for achieving sustainable future of Grapedia. And finally, they have also helped us on how to pitch investors. Thanks to this session, I personally learned that I'm very bad at pitching, but I also learned that the correct message opens many doors. So this is an important thing that we all as a community need to also learn how to reach the industry. Also, I would like to thank the core team of Grapedia. Their participation has been really enthusiastic and consistent. I want to make a special mention to three persons that I call my partners in crime. First one is Camille, the vice chair of this Grapedia CIG. Camille, thanks for accepting this challenge and the support all along the way. I still remember being together in Zaragoza in the annual training school when we got the big news, right? Being here today with all of you present feels like a full circle moment. Also, I would like to thank Anne-Marie Digby, which is, as Mario Pezzotti many times said, the true person allowing all of this to happen. Not only you have been an excellent great holder manager, but also a sister to me. Thanks for allowing me to learn from the best. And finally, Marco Moreto. Marco is part of the core team, and he has been basically the architect of Wikipedia. You have not only been the architect, that you also have been hosting students, hosting a training school, and we will be truly in doubt with you for being the working hands of Grapedia and for giving solution to our challenge. I would also like to thank our sponsors. So to Novatech from Mercier Group for sponsoring both our kickoff and annual meetings. Tomorrow we will have the talk of Olivier that is here present. That he will talk about the roadmap on how Grapedia can benefit the industry. So I'm really looking forward for your presentation. We will also like to thank Igalo Winery. They have sponsored us the US counterpart of the plenary speakers that we will have tomorrow. And they have also assisted or enabled the front-end integration of ChromoMap in Grapedia by financing the travel of Lakshay Anand, that is also going to talk today. And he will explain how we are integrating this tool in Grapedia. And a special thanks to Nick Dukuzlian for his long-standing interest in the evolution of the project. And finally, but not least, to Sequencia Biotech. Sequencia has been funding us whenever needed, not only with Grapedia, but also in Integrate. Sequencia has been a partner since then, and I would like to especially thank Walters and Severino for believing in our idea. Also, I would like to thank other communities and databases that are in collaboration, with strict collaboration with Grapedia, especially genomics.com and Grameen. And we hope that our portal will be fully interoperable with these other databases. And we also would like to give mention to ChromoMap and Nextflow, which are being integrated in the portal. So I just want to briefly comment on how we started the Grapedia. So the idea behind Grapedia came from many years ago, but it really became true on a working group meeting that we had in Lisbon, in the University of Lisbon. It was almost starting the Integrate construction. We were invited by Margarida Fortes in the University of Lisbon, and it was actually the first time I met Camille. And I remember we were on a lunch, and Mario was asking me, so what's next? After we finish the cost action integrate, what's next? And I said that if we are doing everything as we hope that we will, by the end of the cost action, we should definitely move forward for building a unifying database that holds for any type of data, literature-based, genetics, phenotyping, omics, climate data. Why not to dream of that in the future? And now we are here. We're celebrating a prototype that I hope all of you will be able soon to test. And we expect actually from the community, not only the audience here, but all the people who are online, to give us feedback about this portal. So I'm almost finishing. It has been a year of many activities. We have been engaging researchers from all over the world to implement this first prototype. We have so far created dashboard contents and started to build the workflow section. We will hear from Marco and all the students going on short-term scientific missions on how this has been implemented during this first year. Now we also need to implement the services because, as you may have heard before, we need to include services that will allow the portal to be sustainable in the future. And these three main pillars, dashboards, workflows, and services, will be the basal skeleton of the portal. Extra sources of data are needed to be implemented. As you will see here, we have been working in integrating some of this data, but future data will be integrated, and we're going to talk during the whole day about this as well. And for instance, hopefully phenotyping or QTL data will be soon incorporated. Grapedia not only targets the research community, but also the industry. So we are very pleased to have people from the industry here, also online. We will have some talks about how the industry would benefit of the database and how they can contribute as well to the database. Tomorrow, in addition to Olivier's talk from Mercier, we will have the talk of Felipe Gainza from Conchitoro Winery. And they will give a pretty much good idea of how the industry is benefiting from research and how we should all work together. So we actively promote this collaboration and invite researchers to be part of this extensive initiative. Before we started with Grapedia, we established the basis of standardization, of data standardization. And this was thanks to the integrate cost action. I mentioned it before. I would really would like also to thank Mario Pezzotti and Anne-François Adam Blandon for being able to lead this community in the standardization of data and generate all of these resources that, because they are now developed, we can use them and build our database. We would like to make also an announcement to the online community. We hope that these 3 days will be of a lot of interest for you. And we're going global. Now we are in more than 200 countries all around, and we hope that you can see that it's getting global. We hope that we are able to collaborate with researchers all over the world, not only in Europe, but all over the world for the future steps of the portal. And I would like to thank you once again for being here present, and I hope that we all enjoy these 3 days here. Thanks you very much. Okay, so we're gonna start immediately with the first presentation on the Grapedia implementation, and I invite to the podium Marco Moreto from Fondazione Edmund Mar. Thanks, Marco.
Thanks for the introduction. 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 Thanks for the Okay, so good morning and thanks everyone for being here. I have to say I'm really, really happy to be here in Valencia once again. Last time was February 2020, so not exactly the best day to remember. I was going to be better this time. Anyway, I'm going to start. This is a brief overview on what I'm going to talk. Luckily or unfortunately, I'm going to be splitted in between the coffee break. So I hope it's not going to be too much boring for the first part, and at least. So I'm going to talk about the structure of the prototype, so the structure of the whole Grapedia in terms of what is the backend, what is the front end, in the sense that what are the data and how they will be presented to users. As well as what are the services that we developed on top of those. And then I'm going to dedicate a little bit more on the web portal itself, since it's one of the services, but it has its own more complex structure. And then I'm going to end after the coffee break talking about the roadmap and the outcome from the training school, since these two are pretty much interleaved, considering the amount of work that has been done for the community into understanding which are the priorities for the first actually version of the portal. Okay, so I'm going to start from the very beginning just to be sure that we are everyone on the same page. Grapedia, it's going to be a collection of all the omics and not only phenomics and phenotyping resources that we have. They are scattered around the different research labs. And so our goal is to allow an easier way to have data exploration and analysis considering all the resources that we have. We also want to provide tools for performing analysis, such as comparative tools and so on and so forth. And as Thomas said, provide guidelines, continuing the work that has been done for Integrape in the sense of how to use workflows, but not only for analysis and also good practices, all for reproducibility of results. That is for sure one of the main important aspects of the whole implementation. And to me, and this is very, very important I guess for Thomas as well, it's to build a strong, passionate, and welcoming community. Since without the community, it will be pointless essentially to start and build something like that. So really, Grapedia is something that starts from a community, going back to the community. Okay. So this slide, I think I showed it also during the kickoff meeting. I slightly changed it. I updated the logo, and but also I changed it a little bit, and essentially tries to give an idea of the whole architecture that we have implemented so far for the prototype. So we start, as I said, from the community, and we're going back to the community as services, but this is not shown yet. Anyway, the resources that we are collecting will be collected into a database. The way we are doing that, it really depends on the kind of resources that we need to integrate. Considering omics data, most of the time they are file like FASTA file, GFF file, so de facto standards for bioinformatics, but also CSV. They can also be specific or more specific tailored or strange file format. And we are able to integrate all of those within the same centralized database. And this is a normal DBMS, so a database management system. But the part of the centralized part is just one of the aspects of the whole infrastructure. The tool we are using, it's all open sources. One of those is Airbyte, that is, it's a Python tool that it's used to fetch data from all over the internet, essentially, and allows to build a specific structure into the database for those specific data. So it's a very powerful tool since it allows us essentially to modify the way we want, considering all the different file formats that we have to deal with, considering that there are a lot. And the database then, so the centralized database is one part, since I'm going to talk about the federative part, sorry, as well. But the important part is the last one. So the API, that is the application programming interface. So the interface is going to be the only it's going to be the only endpoint that allows users to access to all the data. The data will be integrated in such a way that for the user, it will be easy to access to different data sets at the same time without knowing the actual complexity that lies behind. Okay, so this will be hidden. And the technology that we are using is called GraphQL. GraphQL, QL stands for query language, so it's a query language. And it's a pretty simple, I will say, but I'm a developer, so language to learn. But this is not the point, in the sense that using a GraphQL is not meant for day-to-day use. It's usually meant for people that want to perform specific analysis or for developers that want to develop specific tools for Grapedia. Okay, so don't get scared. You don't need to learn another language for working for Grapedia. But still, it's very useful, especially for developers, since we have only one point to access all the data that are behind that. So just to give you a brief example of what is this interface, so how you can extract data, this is a very simple GraphQL query. Okay, so it's a language that resembles JSON in the sense that it looks like a JavaScript object. It's a graph language in the sense that it's nested. We can ask for fields that maybe contains other fields and so on and so forth. Okay, like in this case, we are just asking, provide me some information about genes where the ID it's equal. Like I'm literally just reading out what is in there. So it's pretty straightforward what's happening. And for this specific gene, give me its ID, the annotation information, but for the annotation, only the gene symbol taken from the gene catalog data sets, and some information about its transcript, but the information I want are just the start and position of exon for its transcript. So the end results, it's again a structure done exactly as the query. So the advantage is that from the developer point of view, is that we know in advance how is going to be the structure of the results that we have. The other advantage is that we can ask exactly specific fields and just what we need for different things without really need to know all the structure. In the sense that this query is hiding the fact that in reality we are querying a lot of different tables at the same time, putting all the results together and then presenting the result as a single unified data model. And again, this is the result. It's just a JSON object in the sense that it's a standard format used in the internet and from developers. So it's pretty straightforward to work with. Again, the idea of the GraphQL interface is not to be used for day-to-day use, let's say, from researcher, but it's a very important point for developers and people that want to perform more tailored analysis. Speaking about the data model, so all the data that are living in the database are somehow, it's what it's called like a data lake in the sense that they are just there and they're not integrated. And the interface itself has the goal to put everything together into one single unified data model that it's what is actually presented to the users. And so these data models need to represent as much as possible all the complexities that are in the database. And when we started to think about how we can model the data that we have. This is something that was started in November during the kickoff, also thanks to the work done by Walter from Sequentia and Marco and Fulvio as well. And we decided that probably the best idea is to have some model that revolves around the idea of gene. Okay, a gene, it's that kind of entity that you can find most of the time in data sets. Maybe it's not perfect as a data model, but at least it works, sorry, most of the time, considering the data sets that we have so far. But we also have to keep in mind that the data model is something that will change through time. It's not something that is written in stone. So there might be new data sets, so new type of data, and different fields to add. So this is not something that is going to be like that forever. It's going to change. So the unified data model, as I said, it's the representation of the data that we have in the database. And from the gene, as you saw in the query as well, we can obtain information related to its annotation, for example. So the gene ontology annotation, as well as the interpro annotation or the gene catalog annotation. Of course, linked to the gene, there's the idea of the genome. So considering its physical position and all the information related to the genome. So the genotype, the assembly version, and so on and so forth. Together with the information about the genome, we have information about markers, and then we will have information about QTL and the different traits that are linked to the QTL. And this is something also that will be talked a little bit in detail later on. And then together with, let's say, the qualitative information about genes, we also have quantitative information like expression information, and from the different databases that we have, network information in the sense that the network is the representation of a specific information that most of the time is gene co-expression, and also DNA binding information, and in our case is mostly DAPSEC information for the moment. These are the resources that we integrated so far for the prototype. As I said, related to genomes, for the prototype phase, we just work with the reference genome, so the PN424 in its, let's say, previous and latest version, even though there is, I mean, I don't know if it is the latest, since, I mean, it's an always evolving work, right? So for sure it's not going to be the last one. And same goes for the genes. We worked for the genes related to the reference genome, and with all the different versions, the one that have been done in the latest version, together with the ones coming from previous genome assemblies like V1 and V2 that are still used by the community. And together with that, we have the gene catalog information for the annotation, gene ontology information, and the correspondence between the different versions of the gene annotation, again, for the reference genome. Related to the QTL, at the moment, we don't have any data, but we have let's say, I would say, really nice data model itself. Again, this is one of the topics that I'm going to talk a little bit more later on. Related to quantitative data sets, we have transcriptomic coming from Vespucci and Wittwitz. And the main difference between the two is the base reference that they're using, and the DNA binding information in terms of DAPSEC data, mostly from the MIB transcription factors, and network information, so information represented as a network, to be more precise, are coming again from Wittwitz, so from the Tom's Biolab data sets, and one gene that is a work that will be presented later on in details from Stefania and Walter. And then we hopefully will be able to integrate even more omics data. As I said, the data model is not something that is going to be the same forever. It's something that will be evolved in time. Okay, so going back a little bit about the database, I explained that we integrated the data using Airbyte. That is an extraction, transform, load software. So it's a software that allows to pick up a file that is somewhere in the internet and put it in a database in a very easy way. But Grapedia is not just a centralized repository. Okay, most of the file of the data sets we are working with are files like FASTA file for genomes and GFF, but other sources are living in other services itself. So we integrated in a semi-federative way, so within the same data model, data coming from the centralized DBMS, the centralized database management system, together with services, other services that are coming from the internet. Like Vespucci itself, it's the expression database that has its own GraphQL interface. So anytime you ask for expression data and it's going to be retrieved from Vespucci, is something that is done on the fly. So it's not stored in Grapedia itself. The same goes for information coming from the ontology lookup service or coming from Uniprot. And we can integrate any other API, of course, that are available, all in the same data model. And the same goes for what I call runtime elaboration from file. And I don't know if it does exist a better way, but there are in bioinformatics, there are cases in which you have to work with huge file and it's pointless to save it somewhere like in a database. Like for alignment file, when you have a BAM file, they are usually really, really big and you just maybe need a little tiny part of that. So you're going to need a specific tool for that's going to look for the index and retrieve just that part. And so this is a sort of elaboration you do in real time using a program to retrieve a part of information. Again, also this one, it's been implemented and it's another way to retrieve information from data sets that can relies on binary file. Okay, so going back to what I showed previously about the architecture, I also have the pointer, nice. I just realized that. So yeah, again, we have resources from the community and then blah, 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 something in between. And then we have our interface that is the only way in which you can actually assess and see the data that are hidden here somehow to user. And then we have the services. And then the services relies on, of course, on this interface. As Thomas said, the services we are working on are workflows, dashboards, and the web portal. So starting from workflows, the idea of a workflow is to integrate the different programs into one cohesive pipeline that has to be reproducible, okay, and scalable as well. But reproducible is really, really important. And the way we implemented that is, again, using standards. And the standard we used are Nextflow and Docker. So what they are used for? Well, Nextflow is yet again another language, but again, don't be scared. It's not something that you really need to learn, since we are going to provide different workflows that will be easy to use. Okay, and the idea is that informatics pipeline are composed of different steps, and that might be really complicated to execute because maybe each step requires its own programs with libraries and dependencies and packages and versions and stuff like that. So what the Nextflow does, it's trying to glue together all the different steps. So it's like one language to rule them all, let's say, and to glue together all the different steps, linking the output from one step as putting as an input to the next one, and so on and so forth. The other important part of building a workflow, it's a Docker. And a Docker, it's a technology that allows the creation of isolated environment. So That means that within Docker, we can create one package, let's say, that contains all the programs with dependencies, packages, different versions of different packages, anything that we need for running the pipeline that is completely isolated by our system. So we don't need to install it ourselves. We don't need to affect or infect our system since everything is going to live isolated from anything else. And then the last part is going to be Grapedia that provide the different data sets that we will need for our pipeline to work. And in this one small example, this is like one pipeline that it's very simple we're working on. And it's like a typical example, at least for me, that I'm coming from transcriptomic, is to perform an alignment considering FASTQ file in order to obtain a count matrix that then can be used for subsequent analysis like differential expression analysis or clustering analysis or whatever. Okay, so the input file are coming from my lab, let's say, so FASTQ file, and I just want to have a count matrix. Okay, so each gene, each sample, and a number telling me how much that gene is expressed for that specific sample. So in Grapedia, it will work with something like that. So once you have the Nextflow file in your system, you will be able, of course, you will need to have Nextflow installed as well. And essentially, once you run Nextflow run and the workflow, so the RNA-seq alignment file, the only thing you need to provide is, of course, the files that you want to align. Okay, so in case of RNA-seq experiment, you most of the time as reads like coming from the forward and the reverse strain, and that's it. This is the only input that it's needed coming from your side, because the other thing that will be actually needed for performing the alignment, it's the genome and also the version of the genome, the genome annotation in order to perform that results, so the count matrix. And these will be coming directly from Grapedia. Okay, so this minus genome and this manus annotation, this will take care about which genome you're going to use and which version of the genome annotation you're going to use. And what is going to happen under the hood when you run something like that is that it will be created for you an isolated environment with all the software that are needed for actually running this. The data will be downloaded from Grapedia, and then the actual workflow will start to execute. And in this case, the workflow, it's very typical. So you clean the reads, you trim it, you clean the reads, you trim it, and you create the genome index considering the genome you are downloading, and then you perform the file, the alignment, and you create the file with the actual count matrix. And all of this will be done, let's say, I will say automatically, but it's not because you need to press a button, but I mean, in an easy way, you don't have to figure it out how to obtain the genome and which version of the genome you might want to use. Speaking about dashboards, that is the other service that is provided. Am I in time?
Yes.
There will be the coffee break, so don't worry. Speaking about dashboards, dashboards are a way to visualize at a glance specific information, most of the time related to gene, again, most of the time. There's not necessarily that, as I said. And you do that using plots, charts, and tables. And people that works with R or Python usually might know like Shiny in R or Streamlit or Plotly, or Dash, that is based on Plotly in Python. And so there are different ways in which you can already do that, of course. And it will be even easier to create your own dashboards if you already know how to create dashboard with R or Python, just by querying the GraphQL interface. Otherwise, we are implementing our own dashboards within Grapedia using React.js. Okay, React.js is a JavaScript framework. It's not meant to build just dashboard. Okay, but at least it doesn't give us the limitation that we will have with these other tools. Okay. React.js is a general framework for building web application, and we rely on that technology for building all the front end, so all of the graphical user interfaces that you will see for the web portal and the dashboard itself. Dashboard will live inside the web portal. And our idea is to build different React components for each of the different way in which you can visualize data and allow for an easy creation of new dashboards combining the different components that we are going to develop. And finally, the web portal. Of course, a portal is like it's a web application, so it's something that is living on the web that brings a diverse and all the general information together in a hopefully nice and consistent look and feel. And the important thing for us is to have access, control for users, and this is for two reasons. The first one is for security reasons, of course. So we need to know who is accessing to the data, and also to be able to provide specific and user-tailored services for specific needs. So again, another part of the structure, but this time it's to show you that actually the real architecture is something like that, and I'm going to explain why it's that. The thing is that dashboards and workflows are mostly meant to fetch data and then visualize data and use that, okay? So a dashboard is usually, as I said, you take the data and you visualize as a plot, as a chart, as a datagram, whatever. And the same works for workflows. Workflows will run on data that will be fetched from the GraphQL interface. On the other side, the web portal, it's a little bit more complicated and will have its own more complex structure, considering the amount of things that you will be able to do through the web portal. So the web portal will have its own backend, okay, that is using a Redis queue system in order to run different tools, okay. This means that you will be able to run different tool and analysis also through the web portal. And this is true for easy tools like blast alignment, for example, or even more complex one like the one provided by Sequential. And the nice thing about that is that the technology we are using is exactly the same that we're using for the workflows. So essentially, every tool that we have running behind the web portal is actually employing Nextflow and Docker. That means that every workflow that we publish can be directly run within the web portal. But this is true in the sense that you will be able to run it in a cloud system for a calculation. So you will have the possibility essentially to download the workflow and run it locally or run it in the cloud. And of course, running in the cloud will be under the payment of a fee. But for people that don't have the computational power for running extensive analysis or maybe with a lot of samples, there will be the possibility to run exactly the same thing in a cloud environment. And the same goes for Sequential specific tools. Sequential is going to integrate its own tools. Some of those are free and some of those are not. And they will rely again on data that might come, of course, from the user itself, from the GraphQL interface, and it will be running in the cloud. Okay, I think this concludes the first part. I really would like to acknowledge a lot of the people that helped me out a lot in implementing the first in working with the first prototype. And so people from Sequential, of course, like Marco Di Marsico, Fulvio Di Vigilante, Pau Lopez Ruiz, and Walter Sanseverino, of course. and also people from Tom's Biolab, that is David and Luis, and Victor, also Victor Garcia from Valencia as well. Okay, I think this conclude that. Okay, yes. So if you have any question, I will be glad to answer. Of course, I will be here also after the coffee break, now that I think of it, and but also like during the 3 days. So if you have any question, we can also talk later on. And I don't know, for people coming that might be post question in Zoom, I have no idea how I'm going to answer that, but for sure we will find a channel to reply to all these answers, to these questions.
Thanks very much, Michael. Let's first see if there's any question in the audience. Dario, at the microphone or here, right? Presenting here, if we know that, for instance, if you have your this for the online.
Jean-Marc, is it on? Yeah. Asking for a friend. Yeah. No, I'm just thinking about incorporation of other genomes beside the PN4024 genome. Are you envisioning this by still using the PN as a common language based on orthology, or are you thinking about using each of the other genomes available as a reference for all your analysis?
Yes. Yeah, but honestly, I don't know if I'm the best person to try to give you an answer. But let's say from a more technical point of view, it will make a lot of sense to have everything related to the reference genome. So like in sense of orthology genes, and in order to have also links from gene coming from different versions of different genomes and how they are linked to the reference. And this is simply because a lot of information is already ready for the reference. So it will be easier, let's say, to exploit that information. But I don't know if I'm really answering your question. Yeah. Because I mean, I do see the limits of this approach.
Shared with them. All the orphan genes that are not would not have access to all the tools that you're developing, and maybe that could fit back to Grape Genomics or other, with maybe more tailored tools that are more independent from the core database that you're developing. And I'm just thinking about. Okay, let's keep talking. Yeah.
Thank you. Does the room?
I think that's how you took Marco. I'm
So the way which we discussed them, it's since we have partnering with the Sequenza, so Walter Sanseverino, and they have all the knowledge and also their tools are already running online. We are going to use actually their expertise in order to have like a cloud infrastructure to work for our workflows. Okay, so thanks, Thomas, for the second introduction. And okay, so I'm glad that now we are all fed and filled with coffee, so more relaxed also. So this second part is going to be more relaxed as well, so less technical. And it's about the roadmap and the output of the training school, since they are very much interleaved, let's say. And I have to say that so the last training school that was held in Fondazione Edmund Mach in San Michele in Trento, Italy, in June, was my first experience organizing something like that, and I was really honored, of course, and I was really happy. As a local organizer, I had the chance to speak with a lot of people, and it was really nice experience. I enjoyed it a lot, and I hope that everyone else also enjoyed as I did. So the training school was not working anymore.
Check.
It works in which sense? Yeah, but I pressed 3 times. So technical problems. Okay, so the training school was organized in 3 days, and during the first day, we gave a pretty much detailed overview of all the technology that that were used for the development of the prototype. So what now is supposed to work?
Supposed.
Okay, I'm just going to ask if you can move to the next one. Okay, thanks. And yeah, so the first day was presenting like the technical part of the prototype, more or less as I did in previous speech, but with more technical details. Okay, talking about the front end, the back end, data, and so on and so forth, how to use that. And the second day was more about new tools that can be implemented and new technologies that might be implemented in Grapedia together with different data sets that may be added as well. And while the third day was let's say brainstorming hands-on meeting where we were organized in different tables and so different groups, and each of us proposed different ideas, considering what we discussed in the previous days about what we would like to see implemented in Grapedia. So in terms of new data that should be added, new tools that should be added, and so on and so forth. So try again, it was working. So in the end, we got, let's say, a list of priority, let's say, of tasks and tools and data set that we really would like to see implemented in the first version of Grapedia. And this, of course, includes all the possible, the publicly available VTIS genome and annotation. So as much as possible, of course, this is, I guess, one of the most important resources that we have. Then, together with that, an updated expression dataset, since the one that we have so far relies on an older version of the annotation. And considering the DAPSEC information that has been developed by different labs, also we would like to integrate that as well. And of course, QTLs. This is something that there's a specific talk dedicated to that. I'm going to spoil a little bit, I hope not too much, about the work that has been done. And of course, together with that, it's very important for us the dissemination part. So give together with the tools and the data sets also all the correct instructions on how to use that in terms of user documentation. So what is available and how you can use that. And of course, even more important, I will say from my point of view, the developer documentation. So if you're willing to get involved in the development of different tools, how can you use and exploit the architecture of Grapedia in order to create your own tools and get those integrated into Grapedia as well. And of course, information related to the workflows. This is very important seeing workflows. They are all linked and everything, but I mean, it's sort of black box, right? So, and we should avoid that. It enforces reproducibility, but it's really important that you understand what's going on under the hood and you just don't press the button, even though we all like just pressing a button and getting the results. So it's important also to have specific information for workflows and how to use that. Then in terms of tools, of course, from a portal like Grapedia, we all expect to have very standard tools like the JBrowse, also like a genome browser, and we implemented the JBrowse 2 genome browser together with Blast. Of course, this is, I guess, the bare minimum tools that you would expect to have. A search tool in order to easily browse and navigate all the different data sets in order to understand what is available and how everything is interleaved. The conversion tool is something that was discussed during, I had a lot of discussions actually related to how we can move from one version to another of different genomes annotation. So from the V1 to the V2, and this is because the evolution, or I would say maybe development of genome annotations. And again, a QTL browser, I won't spoil that much, but this is like a tool that we really would like to develop ourselves. And last, the dashboards that we want to see implemented. This is something, this is an ongoing work. So some of the things have already been done, like porting some of the VitVit suite dashboard developed from Tom's Biolab. So how they have been re-implemented in Grapedia, tools for gene expression in order to visualize gene expression in terms of heat map or other charts, as well as gene network information, again, coming from the VitVits lab or the OneGene dataset. Okay, so since we are really aware that there's a lot of things to do, and we have pretty high expectations, okay, we really would like to keep the difference between the expectation and reality pretty low. And so that is why it was really important for us to work with, we didn't expect that, right? So I was too serious so far. Anyway, so, we are aware that we are ambitious. Okay, there's a lot going on. There's a lot of things, and we really would like to keep the difference between the expectation and reality as low as possible. And that is why it was really important for us to work with a prototype. Okay, so having, let's say, a safe environment without anyone to watch. No, let's say a safe environment in the sense that something that we can use to test out ideas, and I mean especially for ourselves as developers. So to see if things are feasible or not in terms of does it make sense to have this kind of data? Is it fast enough? How can we integrate this with anything else? What happens if this is going to change over time? Which is the best way to visualize a specific information? So all these different questions we might have, okay, we tested out a lot using our prototype. And as I said, a prototype, it's work as a sandbox, right? In the sense that it's a playground for testing out things. So you are, let's say, free to play around with different tools and toys, but it's not something that you really want to use in production. So this is not something that users will be able to use now. Okay, so it's not available for everyone. And that is because, as I said, it's a playground. So the code is not ready for production. As we understand things, there are different tests and code. The code might be full of, bugs, I would say. And again, it's not yet ready for production. And in order for doing that, we essentially need the resources in terms of machine that will be able to run the infrastructure that we have in mind, together with clean implementation of the tools. That again, we tested and they are working the way we are expecting, but they're not ready yet. Okay, so now talking about the output of the roadmap, these are the results that came out during the last day of the training school. So the discussion and what we ended up with, deciding that will be nice to see implemented for the first version of Grapedia. So I'm going to go on giving a brief overview of everything that has been discussed. In terms of gene regulation and networks, so the representation of gene as networks, the representation of the DAPSEC data as an extra layer. Of course, this is something that we already have, but having also another layer of information in terms of motif, like promoters or cis elements that are used by different genes, and together with the other layer of functional notation, so not only the gene ontology, but also or CAG or specific for resistant genes. Talking about the gene card, that is like a concept developed in Thomas Biolab. We would like to complement the information we have so far with a sort of phylogenetic analysis, linking the tool with Plaza, as well as linking it to Gramine. This is some of the ideas came out, let's say, similarly in different tables. So some of the ideas are essentially the same, together with the gene family classification, so extra layer of information related to gene functions. And as I said, there was a really talk about the conversion tool. It's really important, considering that we're not all on the same page. We're not always working on the same version of the genome. It's important to have a clear idea on how genes might evolve through time. I don't know if it's the right, I would say, yeah, they've changed. Let's say change through time from one version to another. So like one gene in V1 might be, two genes in the next version or disappear completely. So, it might be disappointing finding out that your gene doesn't exist anymore. So you really would like to see how things are going and so on and so forth. And talking about enrichment tools, this is something that is usually linked to gene ontology enrichment, but we would like to expand that with any kind of term, okay? Also like a cis element or different kind of terms that can be linked to a specific gene, and we can see if a set of genes is enriched with this term or not. Network expansion, well, the networks that we have implemented so far, they are, let's say, statically I would say. So it's always it's already present the idea of threshold so a sort of score for which you can start from one gene and expand your network going broader and broader. So augmenting the number of nodes, so the number of genes that you want linked to your initial gene. And then, of course, having an extra layer information in terms of expression information. This is something that is already possible since of the link with expression data. For all the network representation, we are employing, again, a standard tool that is a Cytoscape. Cytoscape is very used from desktop, but it does exist also with as a JavaScript package. So it's very useful since we can represent the same information with the same format. And so it will be easy to essentially download the analysis that you see on the website and keep on going with the same work locally with the desktop application, maybe adding extra information on your own. And then we also discussed about analysis topology of the network. So see if there are clustering employing different algorithms like the MCL topology algorithm. Talking about QTLs, again, I don't want to spoil that much since there will be a talk dedicated to that from Silvia I think during the last day. Silvia and Laura, yeah, they're going to talk during the last day. We had a lot of discussion with both of them, and they really helped out a lot helping out in terms of building the data model. So understanding which is the best way to represent that information, which are the mandatory fields, and how can be linked to everything else we built so far. And some of the points that were highlighted during the discussion with that table were related to reliability score. Since we don't have one single repository for QTL information, and this is something that needs to be collected, we need a sort of a score to understand if it is reliable or not. And talking about queries, this is something that, let's say, emerge from the data model itself. So on how the data are modeled, how we can perform different queries. And the way you want to do is starting from a trait and going to the position that might be physical or genetic position, and the other way around. So considering like one position, which QTLs I might have in that specific positions. And also speaking about different position, different regions, perform a comparison between across different genomes. And another thing that was discussed is, again, since the information is very scattered around and it's mostly on papers or supplementary material, it's something that needs a manual curation. So one idea will be to go on with the organization of an hackathon where we can put together people working manually for manually curating the QTL information that are present already in the literature. And we can employ buzzword of the moment, so artificial intelligence, in doing that. And this is like not really a joke, but for sure it will help us out in trying to narrow down the search that we need to do in order to understand which information might be feasible to include and which are not. And finally, a lot have been discussed about the way in which we're going to visualize the thing, because all in all, we need something graphical to actually see things. And of course, we can employ the JBrowse that is already in place, but we are for sure going to use a Chroma map that is a package that has been developed from Lakshay, and there is a talk dedicated to that. And Chroma map, it's very suitable since it gives, let's say, a more we can play around a lot with that tool. So have like a nice representation, more static representation, but with much more layer information. And one of the information we want to give on top of that is, of course, gene expression information from the Grapedia database. And this is a part of the data model. Again, I don't want to spoil too much, but it was really nice since this is again the output from discussion from Sylvia and Laura, and this is just part of it. But there's a lot of information that we are going to collect and how they've been organized and which are the very important fields. It's something that has been a nice subject of discussion. Last part that I condensed, into one slide. Am I in time? I think so. It's about epigenetic and comparative genomics. And so we would like to see a tool, a sort of lift tool in order to easily convert location from different genomes. So for the same version of the same genome, and also considering all the other genomes that we will have in Grapedia, together with information about the histone modification gene family and again transposable elements. So these are all mutation information that we might have. And together with that, again, layer on top of those expression information, but this is something that is pretty straightforward since this information is already present. Going back to workflows, one of the idea was to provide together with the workflows that we are already working on, one that will easily allow people that develop that ultimately a new genome and perform a new annotation, how that can be easily linked the reference genome. So providing a workflow for calculating orthologous genes from that specific genome against the reference genome. This will be really useful since it will be easier to have a link to everything else. I guess this is partially linked, actually, linked to what Dario asked before. And then, of course, having also, again, a link with the phylogenetic information, we can exploit the work that has been done from Grameen and especially their gene tree information, linking directly to their resources. And again, talking about visualization, since we are considering a visualization, genome-wide visualization, again, we will have JBrowse and Chromomap. Okay, so what is the road towards the version 1.0? Okay, so the way I see it is that we need to move from the prototype to the version 1.0. Okay, so we cannot simply open it up since actually it's not running on the architecture that is meant for the amount of queries and traffic that Grapedia is supposed to have. Okay, so we will need to move everything into a more suitable architecture. But the first step for sure will be to move the old database and the GraphQL interface. That at least give us possibility and to open it to everyone. That at least give everyone the possibility to start and query the data and understand what we have in the database. Okay, at least for bioinformaticians, getting used to the interface and performing the analysis, already exploiting what we collected so far. Then, of course, we will have workflows, since workflows are they're pretty, let's say, straightforward to implement and to publish. And essentially, they already exploit the information that is present in the database. So this is something that can come just right after. And to me, it's really at this point important to have a lot of documentation for both of those in order to have people to understand what is the foundations that we're building. So the documentation related to the database itself, the structure, the data model, and how to use the workflow. And then we will have the first version of the web portal. Okay. This will be, let's say, an early access. Well, one nice thing is that, let's say that the release process is something that has to be done step by step in order to have also a constant feedback from the community in order to understand if things are working correctly or not. More or less the same way that we did internally with the prototype. Okay, so testing out things, and we can go one step after the other. The web portal, it's we already of course have like the backbone and the overall structure. And the early access to the web portal will bring the JBrowse and the Blast tool, since this is not something that we need to develop. They are already there and they are pretty easy to integrate. Actually, we already did. And at least we have, the very basic tools that you would expect from that can be used with all the genomes and genes that are collected already. Then we will have dashboards. Okay, that again, they are already developed in their prototype phase, so they just need to be cleaned a little bit, and then they will be available for everyone. Then we will have the sequential tool. This is, of course, also depending on the different time schedule, but I for sure see, as I said in the beginning, we are going to have free tools from sequential, and then more tailored and user-specific tools that can be added later on. And then last but not least, the QTL browser. This is going to be hard in the sense that it's not something that exists yet. So we know how to do it. We have the structure. We don't have the data yet, but we have the technology, and we have the idea on how we want to do it. The problem is it's not there yet. So we need to develop this, and so that is why it's going to be like the last step before we will be able to finally release the first version. But again, all these steps will be always available from the very beginning in order to collect as much feedback as possible. And I think this concludes my talk. I really would like to thank, these are all the people that participated to the training school. I really would like to thank a lot, all of those. I'm not going to read all the names, of course, I'm sorry. There are a lot, but really what I presented is not coming from me. It's all the results of a very nice discussion we had. And again, I was really glad that everything worked out perfectly without major problems, but also like the output of the last day was really heartwarming, I would say, because I see a lot of people really involved and willing to give ideas and suggestions, believing that what we are actually trying to do it's actually doable and feasible. Okay, thank you all. Thank you all. And again, if you have any questions, I'm here.
Thanks, Marco. So we have a couple of questions from the audience, Dario and Pietro, and I think we have a lot of questions chatted, right?
For so people online can hear.
Okay.
Yeah.
So, I'm very repetitive here. I'm so happy to see that QTL browser implementation. It will be fantastic to have. However, we're going further even from the species now. So most, if not, most QTLs are from interspecific crossing. So I think we need to be very, very careful when we present QTL, because based on the literature, most of the QTL when reported using the PN coordinates, using the PN physical content of those QTLs, you're missing the genes responsible for the trait because those genes come from the wild species. So I'm thinking about, since this is still in a vision stage, I think we should think about how, as part of the manual curation, you can include whether possible, if the genome is available, the orthologous regions in the species that are sources of those traits. And maybe at the window, you link and you have the flanking markers, coordinates based on PN, and then port those into the species that is source of the trait. I think the literature is still, there are still plenty of papers suggesting the gene candidates based on the PN gene content, which is often not the case. So I think, yeah, I made a point of that. Let's think about that. I think is the time to think in those terms. Yeah.
I'm glad you found a question.
Yeah.
Is a voice that
Yes.
Okay, so I'm glad it wasn't a question and it was a comment, so I can say I agree. And actually, yeah, when we were talking about the SNP lift, is exactly to have a possibility to have the same region across different genomes or different versions of the same genome or different genes. So definitely we are going to have that information that will be easily shown up together with the QTL information. So thanks again for the suggestion. I think you have to wait a minute. Otherwise, global people won't hear us.
Very nice. I had a very similar question, so I will add what other comment on top of that. So it would be very useful because there is not so much data and the data are confounding to in this QTL browser to have a possibility to estimate also the uncertainty about, for instance, data that are suggestive but are not they don't have a p-value, and also that is statistically the necessary statistical power to rely, but perhaps this to show also data that are suggestive may allow meta-analysis on some loci to get and to be able to even to plan experiments to pinpoint data. So I think that if and also perhaps to add even comments from researchers on those regions, that would make very informative about something that could grow and get to the right target. It's just
thinking about something like Wikipedia where people can place a comment.
Records, say, but also if there is a way to have data that are not reaching the statistical power, but can then later combine it with other data, that would be a big service, because at that point you can really build up knowledge on that.
Totally agree. Actually, one of the discussion was about having a sort of reliability score about all of those. But yeah, maybe doing something like Apollo, so giving users the possibility to add some comments. This is something that is, of course, is totally doable. Considering also that you need to be registered in the platform in order to do that, and so at least this might save you from, you know, unwanted comments. During.
Your historical data versus data that's coming, and how do you weight that? And how do you weight that in your consideration of how you're going to coordinate and engage the community? So when you're talking about historical QTL data versus, for instance, data that might be coming out in the future prior to publication, have you been thinking about how you would work, how you would triage or work with?
I think I don't understand exactly the question.
I think should in a we are dealing with the period I think it would be a very good opportunity to thinking in a web submission portal within Grapedia, right, for QTL.
I can come over here.
Well, basically repeating, yeah. I think that we are already doing that with some resources, specifically the literature-based database that is being incorporated in Grapedia. And yeah, the idea would be to have like a submission form for QTL or eQTL data. I think that is something that we should think of. That's a very good idea, actually.
Thanks, Dinesh.
Thank you, your talk. Question or maybe a suggestion regarding the SIG data. Yeah. And unfortunately, I didn't attend your last meeting, so I already thought about that. But as far as I understood, you plan to provide the SIG data as a sort of network, network or something like that.
Which you can present. So to give like information related to something that is linked to a gene, but is not the only way in which, of course, you can represent the information. Another way will be through like, again, the genome browser or the map where you can see it, you know, along the chromosome. So the different position of the peaks. And
okay. Now, because I was thinking that, you know, experiment open provide 1,000, 2,000 of single transcription factor. But I don't know, I'm getting more and more convinced that the biological question we want really to answer is not which are the target genes for a given transcription factor, but based on a specific genes, which are the transcription factor that regulate that genes. So I was thinking maybe you could think about interrogating for a specific gene, and based on the database, provide all those TFs that regulate the gene or that show peaks in the promoter region or other genomic features of the region. Maybe I'm thinking about, I don't know, possibilities to select the gene select the 1 kb promoter, 2 kb promoter, 5' UTR, and you end up with all the transcription factors that are actually that seem to regulate the gene based on the DAPSIC data.
Totally doable. This is exactly the kind of suggestions we are looking for. So of course, we cannot come up with all the possible scenarios, and having a specific biological question is always a good starting point for then designing the tools that try to answer that. So thanks a lot for the suggestion.
There is something already in.
Remember, micro.
Yeah. There is something similar to what you suggested right now, very recently incorporated in the database. In the Gene Cards app, you actually, when you travel around the different genes that are present in the genome, you can see, based on the few data that we have right now, but hopefully when we have more data, for instance, the one that is generated in your lab, you can see actually the TFs that are bound to that gene. So Luis will show it this afternoon in the demo. That's already incorporated.
Thanks again, Thomas. Other questions?
Yes. There's a question in the chat.
Okay. Do you want to read it?
Yeah.
It's another.
Hi everyone. I was wondering if you're considering to include tools associated to GWAS analysis.
Well, again, this is a suggestion. So definitely, if we have the data, and it's totally doable, absolutely. So this is not something that we thought at the moment, but for sure, again, we have the complete liberty over that. So, and we're looking for not only suggestions, but also people working on that. So I don't know who posed the question, but anyway, it's like if you want to get involved, not only as suggesting or providing data sets, but also like you have already a tool that you developed or you think might be nice to have implemented specifically for Grapevine. Well, we are absolutely open to try and have it to work within Grapedia. So even though we didn't, just to try and answer, even though we didn't spoke about that specifically, it's totally doable. It's done.
That one's from before.
So thanks again, Marco, for the roadmap presentation. And now we are actually moving to okay, so we are now moving to the scientific missions of the Grapedia CIG period. So we have had some students moving around and going from one institute to another to perform some specific tasks related to the implementation of the portal that Marco has just shown. So we're going to start first with Victor Garcia from Aurelian Bombarelli's lab in IBMCP here in Valencia. Please.
Thank you. I'm going to talk about how the community data is got integrated into the. I am Victor, and I'm going to talk about how the community resources were integrated into the database using Arbeit as a technology. So, like Marco has said before, the Grapevine scientific community has done a lot of scientific efforts in the past years, and thanks to that, they have generated a lot of knowledge, data, and resources, scientific resources about Grapevine. Even with the efforts of to standardize this data, these resources are at this moment disseminated, scattered around the web pages and databases. So one of the main goals of Grapedia is to unify and integrate all this data in just one database. No, this doesn't work.
No.
So, how is that good? Exactly. Well, we have talked before from the GraphQL API and how the data can be queried. And what I have done for the project was using the algorithm technology to get all those results. The is scattered and with each one of them their own format, and then integrated into the Grapedia database. What is Arbeit? Well, Arbeit is a technology that uses the principle of extraction, transform, and load. The thing is that in one of these technologies just extract and then transport this data before loading into the database. In the case of Arbeit, this transform part is just optional. Not for all cases because we have, if we need a proper database, so we need to transform that data. Just it's because sometimes you can see a TL in the system. So how Arbeit works? Arbeit works using some component that is called connectors. There are two kinds of connectors. The source connector that is extracted from the sources, maybe from the data warehouse, some with some server, whatever. Or you can load this data from every place. And then the source, the destination connector is what is going to get the data that the source connector emits and load it into the proper database that we are going to use. Also, so as I said, the transport part is optional and you can see it. And in our case, what is done is to normalize this data into a proper SQL or relational database format. Well, I want to talk about technical things about the connectors and Arbeit. Arbeit serves these connectors as a Docker images. You can just customize every aspect of these connectors. Okay? The connectors can be coded in Java and even Arbeit. That's the Arbeit can produce for you the templates of these connectors, so you don't have to do the scat. And when I was preparing this presentation, because this connector was done last year, in the last month, like 3 months ago or something like that, they have implemented in the web user interface the ability to do these connectors without any coding. Just go to the web interface and you can do your connectors. So if you want to try your labs to do something, you can just without any extra. The most important, some specifications about these connectors, but the most important thing that I want just to remark was the stream. The stream is how the data is stored. How the data is it's the molecular model with which the data is going to be extracted from the source. Means, for example, you have a GBFF, you have some identifications, start, ending points, whatever. And you don't want all this information. You can model this information and get only the fields that you want from that GFF, or just make combinations of the name or whatever. So that's important. You don't have to get redundant data or excessive data that is not going to be used for anything. Also, these connectors have the ability to update the update algorithm that is already stored in the database. You can just overwrite the existing data. You can upgrade the data if you have a new version for a GFF file, for example, or you can just do incremental upgrades. So for this, for Grapedia, we have developed connectors for the more usual formats. It's FASTA, GFF, GovTerms, but also we have developed connectors for the Grapevine community from formats like integrate reference gene catalog and one gene output. Right. Well, about the quality assessments, is that we have created this method we created. The proper iBite provides their own text of the streams are properly configured, and also we have done or test with sampling. So doing that. So you have created your connector, and then you can go to the web user interface, and here in the top is an image of who will be the you in this input, and then you have connected that and you have connected the program this part is the terminology. You have connected it to a Postgres database. So a destination connector that is a Postgres database. And then you have here a button that is SIG now. When you press that button, all the things that is in the schematic below is what happens. You just get the data from the data source, and then that database, the tree, you can have more than one stream. I mean, more than one different configuration for the fields that it could be get from the data source. And then with all the streams, you get a source catalog. And the source catalog is going to just emit the data to the destination. It's JSON, they are called, well, the technical terms are byte messages, but are actually JSON, are just data emitted in JSON format. So that data is going to go to the destination, and the destination is going to be normalized and converted in SQL records, Postgres records. Well, I just wanted to. I'm not going to get inside the how is the connectors developed and things like that because it's not. But just I wanted to get some considerations when I tried to use this connector to actual real data from Grapevine. Okay. Grapevine is very good. It's flexible. It's very simple. A very simple tool. It can be used for our needs perfectly, but it has maybe some problems that you have to consider when you are going to do that. For example, it's very important because in the case, for example, of the DFF or load times, it's only going to take like 10 minutes to load the data. But in the case, for example, of one gene output file, it's going to take like 30 hours or more. And this kind of processes cannot be, at least as far as I know, it cannot be parallelized because you're just emitting data into a Postgres database. So if you are going to do uploads of this kind of data or upgrade this data, it will be a major problem that will be. Also, well, before Arbite, the transformation part is optional. So when Arbite gets this data, just stores it with one format inside the database that you are creating. This is not a problem when you are going to load a GFF. When you are going to load, for example, again, one gene or integrate catalog, it could take dozens of gigabytes of hard drive. So if you and that data doesn't disappear from the hard drive automatically. You have to start manually maintaining that part of the byte. And then it was just to highlight some one consideration. Like the thing is that we are using relational databases, and I understand why, because it like the most just about relational databases. Maybe the people here knows databases, and it's pretty easy to develop with a relational database in mind. The thing is, for example, just for loading the GFF file, we are introducing inside the relational database more than the 100,000 of records. So if you are going to load the GFF file, you have to start manually maintaining that part of the byte. So if you are going to just upload one GFFF file, it's not a problem. If you add two, it's not a problem. But in the future, if is constantly expanding and more data is going to be deposited in the database, the national database systems are not prepared for this kind of this volume of data. Are prepared for more or less not. It's not very small. It's a good number of records that could be in a database, but it's prepared for a moderately bigger or moderately size number of records on database, and for high to low. So is that a problem of use for this? So what I mean with this is that if you just to depositing data, and don't process stuff, it will be that in a moment in some future, sometime in the future, when users are going to just do query to this data, this annotation for this gene or whatever, they are going to experience a very slow time to get this done. So from my English is broken, but what I mean is that the slows are going to the queries are going to be pretty slow, and it's going to degrade. And I'm not a doomer. I mean, this is maybe I don't know. But the only thing I want to say is that we could consider maybe complete the distributional database to a non-relational database. Could solve it in part these problems. Okay. And I have finished. It's not a technical thing we wanted to work at. I just wanted to acknowledge it to Sequentia because they have hosted me in the room when I was developing the connectors. Marco Moreto that has designed this architecture, with impressive, and has aided me in the development of the connectors. So. Thank you very much. The scientific missions that are going to be presented, we are going to leave at the end time for so we move immediately to the David Navarro. He's a postdoc in the Institute of Integrative Systems Biology here in Valencia. He's the integration in Grapedia and the gene reference data. David? Okay, perfect. No. Okay. So, hello everybody. I hope you hear me well. But today I would like to talk about the resources that we have developed in our lab and the way they've contributed for and during my short-term scientific mission in FEM with Marco Moreto. So essentially, I'll be talking about three things and how they contribute to different parts of the full stack database, which is Grapedia. So we've got first the Grape gene reference catalog, which we developed during the last cost action called Integrate, and how we've incorporated this in the database of Grapedia directly. Then we've got the correspondences and equivalences between the different genome annotation versions and how we have decided to tackle this issue, at least within Grapedia at the moment. And finally, the gene cards app, which was first developed for our visualization platforms, and then I've been implementing it in Grapedia during my short-term scientific mission in March. But firstly, I will start talking about the catalog and how that journey started. So firstly, this came up as a necessity for ourselves to interpret omic data.
So we started looking through the literature and coming up with like essentially a table or a database which includes gene symbols and gene functions for over 2,000 genes. Initially, we started with secondary metabolism, and then we opened it up to other collaborators, so we would go into other domains of Grapevine research as well. We currently have it in the VCOS v3 genome annotation, but we are planning to move to the new T2T annotation once it's ready within Grapedia. We share this resource in the website, so you can access it through this link if you want. It's essentially very simple in a way, but one of the main features of it is that we've got unique V3 gene ID and gene symbol pairs. So we've tried to assign every gene with that we're adding to the catalog with a function and with a functional validation level as well, and citations to journal articles where relevant, right? So if we focus on this, we came up with a system of like 6 levels of evidence, where on the lower end of the spectrum, we have basic evidence based on homology searches or other equivalent algorithms. And then we've got on the other side of the spectrum, experimental evidence that in the case of Grapevine, because transgenesis is quite difficult, we keep on with top marks still certain enzymatic characterizations done in other species. So we've made this scheme with limitations of Grapevine as a model in mind. So in order to move to the T25 version of the catalog, we have provided like a submission form also in Grapedia where anybody can submit their genes of interest and the gene symbols they want to give them, as well as the description. So it's pretty straightforward because the compulsory fields are in yellow. And some of them are conditional, so depending on the level confidence you assign to a particular function, you will have to provide the extra data. You can find this in Grapedia, and you're free to email it to the Grapedia email address, and we'll hopefully incorporate it into the new catalog version. Soon we will announce, like just before the end of cost action, a short workshop, maybe of a few hours, for anybody that wants to ask any questions on how to submit genes or wants our help towards doing that. And well, this brings me to a bit of the basics, but I want to stress the difference between gene models and genes as a concept. This is something that Marco was mentioning, but when we go from one annotation to another, we may change the gene model, but we might still be referring to the same gene. And even though this seems quite straightforward, it's not so simple in reality, right? So we've got the gene IDs, which in Grapevine have for most of the assemblies been unique, let's say, for each annotation, and they can help bioinformaticians at least to identify like at what time was that work done, because they will link to a particular genome annotation version. And then we've got the gene names or symbols, which can be used to trace a particular biological function, regardless of the assembly or how many models represent that concept. So here I wanted to show a simple example with the lacase 40 gene, where we see that in V3, 2 models have then been merged into one in the V4 annotation. We have this kind of issue and many others, like splitting genes. Some genes might disappear. Well, hopefully that's not very good, but it happens. and others will appear because more of the genome has been discovered, let's say. And in our case, like I think it's better if we provide all the different gene IDs, but we make it very clear how to go from one to another with the limitations that you need to be aware of that problem, right? Like it's not so easy to say that two gene models are the same between annotations. So we've also thought of Grapedia as a resource to inform the community about Grapevine Genomics, right? So one of the efforts is to show the slightly complex history of like genome assemblies in Grapevine and the annotations that go along with them, as well as when those annotations have been transferred to more recent assemblies. We can see that here, like some of the IDs are recognizable and they can directly be associated to a genome annotation version, but it's not always the case. So in this regard, I wanted to talk about Liftoff, which is a tool we've been using because it works quite well to transfer or the whole genome annotation to another assembly. And it does them quite well. And uses Minimap behind the scenes, and it searches for the single best hit for every gene in your annotation file, let's say. It can also be run in copies mode, and that means that if there's a region that none of your genes match with their best hit, but still some genes might hit there with a second hit, let's say, that is considered a paralog, and it's something I think is worth doing because if we've got a set of curated genes and these are not picked up with in silico annotation strategies, we could add them after like this. So here is a typical example that you can see in a genome browser where we've got 4 or 5 different annotations assigned to the same genome, in this case V4, and we want to answer the question of like which models are like referring to the same gene, let's say. So if we're looking at a JBrowse, maybe you will all agree that this gene, for example, even though it's mapping to the same region in V4, doesn't look anything like this 8X model, but all of these, even though they're different, they sort of seem to mark the same gene, right? So we came up with a system to compare overlap of coordinates between different annotations and also their subfeatures. And we compared them in multiple, we come up with a semi-quantitative way of like classifying equivalencies. Because you see, like a gene is not simply the coordinates of the gene. Then you've got subfeatures and maybe different transcripts, even different CDSs. So that's the same. To this we have also added a metric like of synteny. It's very simple, but the idea is that we can be more sure of a gene model equivalence if during liftoff the neighboring genes also were lifted in the same order, let's say. So if the neighboring genes are the same after liftoff, then we give an extra level of certainty. And if either of these are not the same, then we say that the synteny has not been conserved. Of course, there will be false positives and even false negatives here, but it's something that we can provide as an extra information for particular gene equivalence. And yeah, the system I think we think works sort of all right because in this particular example, the genes that we would, I don't know, like judge as not equivalent end up being discarded in this automatic system. So instead of looking gene by gene, we can do a single like operation and have all of that output it in a table. So here we've chosen, we made a score from 1 to 11 to provide like better, so where the gene model changes less, we've got like a score of 11. but still we consider that like at level 6 or so, like we're getting sort of like what you could interpret as like a similar, like something referring to the same gene. So this is what we've included in the Grapedia database at the moment. We're not including these very low scores because I don't think they're worth mentioning or sharing. But still, I think we're planning to share the metrics so that if you've got a low score, or let's say of a 6 between two gene IDs from different variables, then you can take a look and see it yourself, basically, because it may or may not be referring to the same gene. And at the moment, we're holding equivalences between all to V4. Will for sure will also include V5 once it's ready. And yeah, and finally, I'm going to talk about the Gene Cards app, which within the visualization platform, and how we have moved that to Grapedia. So there are two aspects here, like the data that we have in BitVis that we ported into the Grapedia database, and also the front end, which we've tried to replicate. At the moment, it's in our Shiny, but we've moved it to React JavaScript. So essentially, our main source of data in the BT's visualization platform is transcriptomic data, and we try to download as much as we can from SRA studies that are done with Illumina, because this is the filter we chose. And at the moment, we have almost 5,000 SRA runs that we classified into tissues, but this is the general pipeline where we download and then end up like with count matrices. Of course, it wouldn't be possible without a high computing cluster, and that's why in our institute, we should acknowledge that we've got Garnacha here to help us. We've now improved this. It used to take us like around 2 to 3 weeks to run this whole pipeline for 5,000 runs. And now it takes us about 2.5 days. So it's like 7 times better. So this will be good news for Grapedia in the sense that we can run like more frequent updates. And indeed, like the publishing of results, like in terms of transcriptomic studies, is like growing like dramatically in Grapevine. We used to, in 2021, a query like found 4,800 studies, and just 2 years after, we almost have, I don't know, 9,400 studies. So it's doubled in the space of 2 years. It is good news for us because in our Gene Cards app, we mostly had evidence from fruit and leaf and a bit of flower maybe, but not much could be done in terms of like co-expression networks for other tissues. And it's something that now when we classify the 9,000 runs, I hope we will be able to offer more applications. This is an interactive plot using Plotly, and if you hover over any data point, you'll be able to see the SRA ID corresponding to that expression value. And this is useful because you can explore a particular gene and how it's expressed throughout different experimental conditions. We allow input of both gene symbols from the catalog and also gene IDs. So in this case, I'm showing you MIC2, and it also has all this information retrieved from the catalog. But we can also input any V3 gene ID and also get the same expression data without, of course, the functional data, except for an automatic annotation term. So yeah, the aim was to implement these gene cards in Grapedia. So with that in mind, I went on to FEM like in March, and during those 2 weeks, I really enjoyed like learning about how front-end development works in JavaScript, and we implemented the VitBis gene cards in Grapedia. We also had this tinkering with Grapedia training school that Marco described, where we got very useful feedback on how we should carry on working on the front end. So this is a snapshot of the result. we came up with a box.plot module that should work as an independent component and basically be able to plot data from VidBees, but maybe also from other databases. So that's the federative notion we've got here. At the moment, it's just showing the same data that VidBees has, but this will not be the case in the future. We've also shown the gene catalog information here. And we plan in the future to provide links, as maybe it was also mentioned, to other external sources, such as Grape Genomics, where in the future we'll be providing equivalencies to other genome and even other species, because this lift-off system works well between closely related species, and we can repeat this and provide equivalencies to the annotation we want, I think. And this will help us link to other J browsers or whatever we want to do in terms of coordinates or transferring. So yeah, I would like to thank you all for listening, and also special thanks to members from my lab that have also participated in what I presented today. And a special thanks to Marco Moreto for my stay in FEM and how I learned a lot in terms of web development. And thanks also, Anne-Marie, for all your help organizing Grapedia.
Thanks, David. I think that we are now in very good timing. So the next and last presenter of the morning is Luis Orduña, and he's gonna present the gene co-expression network models in Grapedia.
Okay, so thank you everyone for being here. It's a pleasure be describing what I did with Dr. Marco Moreto in FEM. I know that we are running maybe a little bit just on time, so I will try to be as brief as possible. So as you may know, in our lab we are interested in the study of the secondary metabolism, which is controlled basically at transcriptional level by transcription factors. Because of that, we built in our lab gene co-expression networks that were used as transcription factor function prediction tools. So just as a brief introduction, a gene co-expression network can be described as a network that represent where the nodes represent all the genes present in a genome, and the edges depict co-expression between genes. These networks are usually ruled by the guilty by association principle. That stands that if two genes are co-expressed together, it's quite likely that they will be sharing the function, or they will be involved in the same function. Gene co-expression networks, as most of the biological networks, usually present scale-free topology, which means that we will have a lot of nodes that will have only a few links, and then we will also have a few hubs that will be largely connected. So in our lab, we believe that the gene co-expression networks can be used as a proxy of regulatory and biological networks. So just as a brief introduction, when you build gene co-expression networks, you can decide to use supervised networks or unsupervised approaches. In first place, regarding the supervised methods, it's important to mention that they are methods based on machine learning approaches. Because of this, supervised methods give a higher prediction accuracy when compared to the unsupervised methods. However, this higher accuracy is only yield when the networks are small, meaning that they contain as maximum 10,000 genes. If the inference problems is bigger than that, the prediction accuracy decreases really quickly. Since these approaches are based on machine learning methods, they also require prior knowledge to the system. In this training process, the supervised approaches also require a lot of hardware resources to perform the training. Because of these inconveniences, the supervised methods are not the best method when you want to build genome scale networks, and you can only use these approaches if a feature selection step is performed in first place in order to reduce the inference problem. On the other hand, we have the supervised methods, which are built on the guilty by association principle, and they do not require prior knowledge because they are not based on machine learning approaches, so they don't need to be trained. These unsupervised approaches allow us to analyze genome-wide inference problems, and this is very important. They gain robustness the more samples are analyzed, meaning that the more studies that are incorporated to the networks, the better the prediction accuracy of the network will be. Because of this, we decided to go with the unsupervised methods for constructing our networks, and within when we decided to go with the unsupervised networks, the first thing that we did was going to SRA database and downloading all the RNA-seq datasets that were publicly available in this database. We decided to go with RNA-seq instead of microarray technique because, as you all know, RNA-seq allows to detect the expression of all the genes detected in a genome, and microarray only is able to detect the gene expression of the genes that are present in the chip. So in total, we were able to download 131 SRA studies that were first automatically and then manually classified according to the tissue origin. Thanks to this classification, we were able to separate 67 SRA studies that were coming from berry samples, 42 SRA studies that were coming from leaf sample, and in total, only between berry and leaf, we had 83% of all the downloaded data. Thanks to this classification, we were able to generate both tissue-specific networks, meaning networks that were composed only with SRA studies that were coming from berry or from leaf, and we were also able to build tissue-independent or condition-independent networks with all the 131 SRA studies that we downloaded. Here you have a table with a summary of all the SRA libraries that we downloaded with the automatic and then also the manual filterings. So within the unsupervised approaches, we designed two different pipelines. On one hand, we have the aggregated approach, and the other hand, we have the non-aggregated approach. I don't want to get into the details because it will take a lot of time, but as a main idea, you have to know that the aggregated approach is based on building one network for each independent SRA study that we downloaded. So for instance, in the condition network, on the second to last step, we have 131 independent networks that we merge together in the last step in order to create the final aggregated network. On the other hand, the non-aggregated approach is based on merging all the information in the second step, and from this point onwards, we build a final network that will also contain the information of all the experiments, but merging the information here or merging the information here makes quite a big difference on the output of the pipeline. So after generating the networks, what we did in first place was assessing the free topology. And as you can see here, the three networks that we built, the berry network, the condition independent, and the leaf network, present free topology, since again, we have a lot of nodes here that are poorly connected, that have a few nodes, and then we have here a few hubs that are largely connected. After assessing the scale-free topology, we assessed the performance of the network. For doing this, we use the area under the receiving operating characteristic or AURO parameter. This parameter basically takes an ontology as a reference, and it compares how similar are the prediction of your network in comparison with the reference of with ontology of that we are using as a reference. If the networks predict, so if we take a gene of the network and we try to predict the genes that are co-expressed with this gene, and this group of genes is exactly the same that appears on the ontology, the output of the network it will have a value of 1, meaning that the network prediction is exactly the same as the information contained in the ontology. On the other hand, if none of the genes predicted by the network are present with the gene that is being assessed on the ontology, the output value will be 0.5, which means that the prediction is basically random. So what we did was comparing the aggregated and the single or non-aggregated approaches using 2 different ontologies, using MAMMAN and using Go gene ontology. So as you can see here, when using MAMMAN, which is the best ontology when working with plant species such as grapevine, the aggregated approach yielded a better performance in comparison to the non-aggregated approach. This also happened in the leaf network, but not in the berry network, which is not very surprising, taking into account that the berry transcriptome is really variable and it can be affected by the transcriptome variability. So since the aggregated approach yielded a better performance in both the non-aggregated and the leaf network, we decided to keep working with aggregated approach networks. So after building the networks, we had to decide how to use the networks as a transcription factor function predictor tools. For this, we generated 2 different strategies, which basically are 2 sides of the same coin. On one hand, we have the pathway-centered network analysis, which consists basically on taking the genes of a particular pathway of your interest. In this case, we're showing different routes of the secondary metabolism, but you can also grab genes involved in other functions such as resistance to biotic or abiotic stresses, the tissue development, whatever you want. And what we do is create a co-expression network of the genes involved in a pathway and some transcription factors that we think that may be regulating this pathway. What you can see here is that the networks are able to clusterize the genes of the pathway with the transcription factor that are regulating those pathways in an independent way. For instance, here we have all the stilbenoid pathway with 2 different regulators of this pathway, completely separated of the anthocyanin and proanthocyanin metabolism, which is here. Thanks to this, we can use the network as a clustering system that allows us to identify regulators of pathways of interest. On the other hand, we have the gene-centered network analysis, which basically starts by selecting a transcription factor of our interest. Once that we know a transcription factor that we want to study, what we do is generating a list of 420 genes that are more co-expressed with our transcription factor of interest. When we have this list, so we are taking 420 genes because the networks were constructed using the V3 reference genome that has a total 42,000 genes. So 420 is roughly 1% of the genes described in the genome. What we do once we have this list of genes is performing a gene set enrichment analysis using the ontology, the MAMMAN ontology. Thanks to this analysis, we can know which functions are enriched within the 420 genes that are most co-expressed with our gene of interest, meaning that we can try to predict the function the transcription factor is regulating. So in order to make the information of our networks available to the Grapevine community, we developed this aggregated GCN app within the Bitbit platform. As you can see, when the user accesses this platform, they can select some networks of their interest. So for instance, if they click in the berry network, you can see here that we have 2 different tabs. Here we have the individual GCN tab that allows the user to introduce a gene symbol or a gene ID in both B1 or B3 annotations version. And the app will generate the 420 genes list expressed with your gene of interest. We have added some new functionalities. So for instance, if the user click here in the Run GSA button, the app will generate the gene set enrichment analysis interactive, and you can click on the dots to see which function are enriched in your description factor against centered network. Then the user can also click in the create proximity clustering plot, and it will generate this plot in which the bars represent the chromosome length and the red dots represent the coordinates of the genes co-expressed with the gene that you entered to the app. So you can try to detect, for instance, clusters of genes co-expressed with your gene of interest, such as the one that I'm highlighting here. On the other hand, if the user click on the interaction network tab, they can also. So from the 1st of May to the 31st of July, I was at FEM working with Dr. Marco Moreto in order to implement the VidVis apps into Grapedia. So as both Marco and David highlighted, I was working in the front end development. So I was working in this part of the let's say workflow. Basically, I was retrieving the information that Marco added into the backend using the API that he explained before, and I was trying to develop different dashboards. The dashboard, as they explained before, are based on React, and they are components based on different libraries such as React, Plotly, and Cytoscape. They are a pre-designed composition of data and visualization that can be easily reused. Well, basically, this is a little bit more of info about React. But the most important thing is that React allows us to create reusable and independent components. This is important because people can help us by creating specific components if they are interested in showing some specific plots or show some specific information. These are the different dashboards that are available right now in Grapedia in the web portal, as you will see later on the Grapedia tutorial. But yeah, I was working mainly on the networks dashboard, allowing to generate a dashboard that took multiple genes as an input, and it returned a co-expression network between the different genes given as an input. So here you have a screenshot of the dashboard. You will see it later in more detail. Basically, just as an example, allow me to tell you that this dashboard works with 3 different components. On the main component is the network's JavaScript that controls the input and the output of the dashboard. So the gene tags with drops on JavaScript is the one that takes all the input given by the user, and it gives the information to the main script. Then the main script queries the backend and gives the information that has been retrieved from the backend to the Cytoscape network component. That is the one that shows that displays the network to the user. And this is basically everything that I have to tell you. So as a take-home message, I would like to highlight again that the structure that Marco has created allow us to create components that are really easy to reuse and to edit. So as a part of a community, just feel free to let us know which components you think could be useful to have in Grapedia. And also, of course, you are more than welcome if you want to build it by yourself. So I would like also to acknowledge Marco for having me in FEM, and also to Anne-Marie as a grant holder manager, and also to all the members of the Tomsville Lab that have been involved in this work. And if you have any question, I will be pleased to ask them. Yeah, if you have any question
To any of us.
Yeah, to any of us. Yeah.
Para cambiar, ¿eh?
Thank you.
It's not a question, it's a suggestion. Like we had a few already, but maybe you can think about later and see Dr. Luis. We should also see to try to integrate a set for the promoter identification is developed in the holiday by the group of Bruno Contreras, but we have invited already, so you know about our project, and maybe sometime we can have a meeting and even not until the one or something.
Absolutely, yeah. So all these ideas are always more than welcome. Yeah. Okay. Yeah.
Yeah, I have a question for Victor. Actually, I was really interested in your suggestion about maybe using not relational databases, but not SQL. So my question is, if you tried maybe something, or you already worked with no SQL databases like MongoDB, and you have some suggestion related to that.
So we can explore with that. We can learn about that. Or we can. I don't have any experience with that. I almost the one thing I don't that I know is that exists, but it's not a problem. If you have to learn about that or and try to implement, we can do that. It's not a problem for me. Yeah, because it's just more a concept, just an advice for the future. I don't know. Maybe the mind that I have said is a problem when you are trying to do things that generate a lot of records in a relational database. But what could that happen? For example, you would do the integrate ref gene catalog, and you just do that one that thing one time, or maybe you upload the one gene and you no, you cannot. Yeah, okay. It was just advice. I don't have a crystal ball. I mean, maybe you are going to in the next years you are going to get maybe 2-3 versions of a reference, a new reference genome. It's not going to be updated like every year or something like that. So in these cases, maybe for the future, we don't need to switch the database system. It's just more what the trend says. I don't know what is going to happen to the future. So if you think that maybe that's actually what is going to happen, that you are going to get a accumulate of records, we can check it. We can see how it's going to be implemented, and I can tell you with that. Actually, it was more interesting because obviously, like what you said is really true.
Let's say thousands of genomes. That is not that deep. It's not likely. I mean, going to explode. So, considering the thing that we might have a lot of relation, yeah.
No. So see, it's going to be a problem.
Yeah, maybe considering that we are going to have a lot of relationship between different data sets, and so yeah, maybe having like a sort of on SQL, no SQL structure, you know, it might actually work. So it's definitely a good suggestion, and now I'm curious actually to try it out. I'm actually curious, and I will definitely try it out to see how it will be to change the backend from finally from Postgres to something else. So it's definitely a really nice suggestion. And if you're willing to help in testing that,
I can get some time to. And so maybe I don't know what we can do. Okay.
Yeah.
Doreen wanted also to ask.
I think this is the question it's all this discussion here.
There are different tools for different things, right? And so the SQL database provides you a tool for integrity and QC and QA when you're having relationships between things, but it'll die on a performance for a web. So as you guys are moving forward and talking about NoSQL databases like Mongo and others, I think you're going to need to think about the different tools you'll need depending upon what you're serving. So if you're looking for a performance-based thing, you're going to have to do that almost ETL again to a NoSQL database to be able to give you the performance that you're going to need for some of these systems, but you're not going to want to necessarily sacrifice your SQL database. So you might be thinking about this now as you're moving forward on that. And I think it would also be value into maybe having a discussion with others of lessons learned. So you might be thinking about those earlier than later on, right? As it's just a comment for you guys to consider moving forward. And then I just want to tell you, it was really lovely to see the scientific missions and how you worked on that. And I'm wondering if you thought about, as you move forward, if you have an opportunity to do more, if you could think about how each group could iteratively build on each other. Like I saw a really nice synergy from the first expression data sets to your co-expression networks that could have been, as each one of your students are doing a scientific meeting, they could iteratively build. I had then a follow-up question. When you did your scientific meeting, did you guys develop documentation for the next person to follow, to build on? And if so, are you sharing that with each other?
Yeah, in some ways, so the code, we added comments where needed. Yes. So the components in this case, I was working together with Luis. So if he needed anything explained, we would talk about it. But yes, we will carry on working on the documentation because we may need more work. Yeah.
Think about as you guys are starting to move a developer's platform forward, that part of your things might be internal code review with each other, because as you do that internal code review, you get good documentation. You get you may not get good documentation, but you get documentation. And that helps like teach the next person.
Yeah, we could think of doing that. Yeah, yeah. It's a good idea, yeah.
It's actually something we are thinking about. Maybe in a close few. Yeah, commenting all the code and preparing guidelines, or just to try to help people that comes later and wants to help on the initiative. Yeah.
Wow, nice. Okay, just one naive comment. Because I think the field is not only basic science. There are companies and so on. They can provide part of their data that they want to share and so on. Nowadays, federated learning is quite useful for data. Particularly, there are some tools. One is developed actually in my department. It's called Flower and is easily available. Federated learning allows to have a sort of different contributions with certain kind of rules that all the actors are sort of expected to follow and to respect. And that could be a way also to influence the mind and allow for more people to contribute with data and knowledge.
How easy is to do it, and how many people would like to be involved on that also? Because yeah, it's something that we think we have to think about in the near future. Yeah, absolutely. Yeah.
Well, I think finally we have a new question from the Zoom chat. Are reference works for the gene function checked for contradictions?
In some way, for the ones that we've already added, yes. But the whole idea is that the catalog is open to community input, so maybe the workshops that we'll announce will be useful for this. But yes, we're gonna deal with this. Like if you think if you want to provide contradictory information, we'll have to deal with it and see where we include both, or like choose the correct one or something like that. But yes, yeah, we were planning on incorporating any input that the community brings, as long as there's like evidence to back it up. Yeah.
Welcome back. We are gonna continue with the talks of this afternoon. We have a couple of more presentations regarding scientific missions that have occurred during this CIG grant period. And to those, we are gonna go into a brief tutorial on the prototype, and Luis is gonna present what we have done so far for this prototype. And then we are going to listen to a presentation of Ana Conesa, and that would be pretty much the whole day. I hope that you had this morning very nice presentations and that you have learned a lot about Grapedia. Okay, so we are now going to the next talk that is going to be a dual talk by Camille Rustenholz, which is the host of this short-term scientific mission, and Antonio Santiago, who worked together with Camille in developing the new annotation for the T2T genome assembly. So I give you the stage. Thanks very much.
Okay, so hi everybody. So thanks Thomas for nice introduction. And of course, I didn't do the CSM. It was Antonio. I was hosting him, and it has been 2 great months. So I've been hosting, but I'd like to use the stage right now first to thank all the organizing committee, because they've been doing so far, and I'm sure they will move on with that, an excellent job for welcoming us and organizing all these great events. And also I'd like to thank 3 persons in particular, Thomas, Marco, and Anne-Marie, because you cannot imagine the tremendous amount of work that they've been doing during this year. So, to make all this become true. So warm thanks to the 3 of them. So before no problem. Sorry, I'm short. Yeah, it's better. So before leaving the stage to Antonio, I thought maybe it would be important to give you some heads up on what's going on with the PN 40024 reference genome, because maybe you've seen that there were several versions showing up at the same time. And so I'm going to give you more details on what we call the T2T genome assembly, that it's out there, and that we are going to use as a backbone to build the Grapedia portal. It is actually not working, guys. Yes. So the T2T genome assembly has been performed using the new PacBio technology, Hi-Fi reads. Was built using a 42x coverage of these reads, and it's a totally de novo assembly. which it was not the case for the V4. And so this assembly has been published in May earlier this year, and made the cover of horticultural research paper. So just a few numbers to show you that the metrics are really good and better compared to what we had previously. So, for example, in terms of especially the number of contigs that were assembled and constitute the chromosomes, it's really a telomere assembly, as only 20 contigs were needed to build the 19 chromosomes. The contigs are now very long; they are chromosome lengths, basically, and there are no gaps anymore. In terms of completeness, so we use the Busco analysis to confirm that this is the PN version that has never been as complete and as contiguous. So it's actually a very nice resource to build a portal on. So we compared the version 4 with the T2T assembly, and what is obvious is that basically the amount of sequence that has been added, it's mostly located at the centromeric part. So really showing that this new technology helped to resolve the repeats. Oh, nice. The repeats in the centromeric regions, and also it helped to identify and correct some previous myths. Also, it helped to resolve integrated structural regions, just like this. So we really think that this version now is the best that we had so far for the reference genome. So we call it assembly, and this is actually true because we were able to identify 19 centromeres out of the 19 should be. But telomeric regions, there are only 2 regions that are missing on chromosome 15 and 70 here. This is because at the end of at the extremities of these 2 chromosomes, there are 40 SR DNA tandem repeats, which are difficult to resolve, and it's even harder to reach the total chromosome. We really believe that after these 45 rDNA repeats, the telomere should be right after. So with this new assembly, we also retrieved the heterozygous regions that we identified previously. So they are mostly located, the largest one is on chromosome 7, and the second largest on chromosome 10. And what we were willing to do, along with Antonio as well, was to test the phasing. So meaning that we were willing to see whether the haplotype was also contiguously assembled. So we tested the phasing, especially in these heterozygous regions. So before going into the phasing, I'd like to remind the community about the origin of PN 40,024. So this is a genotype that has been developed at INRAE Colmar, where I'm working. But I actually started working there. So the original publication about this genome claimed that it was originating from 9 selfings of Pinot Noir, but with the publication that we made earlier this year, we demonstrated that it was not the case, and that PN 40,024 doesn't really have the correct name, as we figured that it's originating from from Helfensteiner, which has been successively selfed 9 times. So it's not completely different Pinot Noir, as Pinot Noir is one of the parent of Helfensteiner, but definitely half of the genome of PN 4024 is not Pinot Noir. So PN 4024 is not that was for sure, but now we definitely know it's comes originally from Helfensteiner. And by the way, I just used directly in our greenhouse the PN 4024 has white berries, either Pinot Noir or Helfensteiner. It's a new genotype originating from Helfensteiner. So, what we identified with the V4 assembly, so it was the genome was a mixture of Pinot Noir and Schiavagrosa haplotypes, so the two parents of Helfensteiner. You can have an overview here of the mixed haplotypes that we identified by then. And where in green are the heterozygous regions. So you can see, like for example, here on chromosome 7, that with the V4 was a mixture of the Pinot Noir and the Schiavagrosa haplotype, meaning that the phasing was probably not correct. Right now, with the P2T assembly, if you look at this exact same region, the phasing is perfect. So here it's only the red haplotype. So definitely with this P2T assembly, we've reached the top of what we could expect with the current technologies in terms of completeness and phasing for the reference genome. So this was for the assembly, and so this was the basis of what Antonio was using when he arrived in Colmar for his STSM time. So I leave him the stage now.
Thank you, Camille. I mean, I'm gonna start with a brief introduction. That since the first Grapevine genome was sequenced along with its own annotation back in 2007, we've been having a lot of different new annotations, even some of them from the same exact assembly. For example, here the 12X assembly, and we've got for the B0, the B1, the B2, even the CBI1, and that's without counting the B3 or B cost annotation that was performed in a reshuffling of this assembly. Now we've got the B4 for the genome. We're working on a one for B5 in the T2T. And each time a patient comes out, we just forget about the old ones. We got rid of them and go all in into a new, improved, and more complete genome annotation. But there's a lot of work that has been done by the community in those previous gene models that cannot be lost, and we cannot get rid of them. So that's why we need those correspondences and those equivalences between the previous gene models in these new annotations. Fortunately, as David already introduced, we've got liftoff that and this tool will be able to map the features from previous genome annotations into the it already has helped us a lot in annotation training schools. We are able to load different different of annotations and compare the models between between those annotations, and thanks to that, we were able to annotate more than 2,200 genes. That number is probably bigger now, and it's getting bigger and bigger. So yeah, and those gene models are not going to be lost. We're able to lift almost 99% of the in IDs from the 4.3 genome annotation. And we have found also 99 comparable genes in total for with only this annotation. In summer, with during the months of June and July, I was invited to Colmar and for working on the genome assembly and its annotation. And together with Camille, we were evaluating and validating the genome assembly, and also working on the reading the annotation pipeline. Our goal is to make the 424 genome assembly the reference for Grapevine, and ensure that it is accessible for the wider community through the Grapedia portal. To help us in this genome annotation, I am pleased to announce that we have a new source of evidence. RNA-seq data is available now specifically for PM424, containing a diverse range of tissue samples, going from very young leaves, tendrils, internodes, young inflorescence, flowering inflorescence, very young berries, green berries, and ripe berries. All of this data, well, all of these samples were collected in Colmar, and they were Illumina sequenced thanks to Daniela Holker's lab, and they were polymerase standard libraries and also RNA depleted standard libraries. And also some of these samples were sequenced by Doreen Werstrap. So with all this evidence, we are rerunning with some modifications this annotation pipeline. This pipeline was developed by Amandine Belt, by Marie Bianca Fogg. And also, well, we are working on optimizing some of the tests, the steps for some of the bottlenecks in the pipeline. Also adding some steps for the new evidence data that we have, and adapting it to the Grapedia standards of first principles with findability, that stands for findability, accessibility, interoperability, and reuse. By now using SnakeMate, we're working on when it changes to Nextflow in the future, and also having the contained containerized in Docker. It starts with the masking of the regions of the genome with the DTI software, and we are masking. We are soft masking those regions annotated by DTI and hard masking them. After that, we have the evidence data coming from protein data coming from different species. Also the RNA-seq data that I already talked about, the stranded, and also the SOC. For the ab initio predictions, the ones that don't have any kind of don't use any kind of predictors, we have different software: Snap, Glimmer, HMM, Breaker3, and GeneID. And finally, we have PM424 4.3 genome annotation listed, so that we don't lose any of those gene models from this annotation. We also giving more weight to the catalog, to the gene catalog genes, and they already manually created gene models, so that they are not so that they are really conserved in the new annotation. Evidence model will be in charge of, with all that evidence, return a final genome annotation that will then be improved by PASA by redefining those the UTR regions, and then once we have a final genome annotation, we can then annotate the tRNAs, have a functional annotation with TCO, with PEG, MACMAN, any kind of ontology. The annotation pipeline will be included as a Grapedia workflow in the platform, so that anyone can use it for their own genomes or any cultivars of interest. And I would like to also introduce you to the IDs structure that will be composed, first of all, for part with the species. The BD part will be conserved, but we are adding the genome version and also the annotation version, so that we are able to solve some of the problems that we have found in some of the previous annotations. We could not be able to some gene levels were changing between the different annotation versions, considered exactly the same. This way, for example, if you find in a publication in a gene ID that it's that you don't know which annotation is it, just by looking at the gene ID structure, you can know already which annotation from which annotation version is it, or which genome assembly was the one used for the analysis. We are encouraging also the use of this ID for other cultivars. We can have at the end of the reference ID an underscore with the cultivar, so that whenever we find any of these gene IDs in publications, we know already that it's a Vitis vinifera gene ID. In other news, I am pleased to inform you that in the Grapedia website, you will have you have already in the button for the downloads to download all these files, the genome, the T2T genome, along with the soft mask one, the hard mask, and also the heterozygous regions. And also, together with my colleague David, we have been setting up a gene browser, for you to explore this T2T genome, where you can load these different annotation tracks from previous annotations, and also a track for the repeat regions, and all the tracks for the RNA-seq data. The RNA-seq data is divided in Illumina and LD-ISO-seq, and for Illumina we have a track for multi-mapping, where we allow reads to map in the regions of the genomes in different parts of the genome. And in the unique one, we only allow the reads to map in a unique part of the genome. And for the ISO-seq, we have the raw ISO-seq reads alignments, and the reads filtered by it is filtered by intern retention variants and pre-mRNAs. And finally, I would like to acknowledge all the members from my team, and also Camille and everyone at INRAE for hosting me there in Colmar. Daniela and Doreen for the sequencing of all the RNA-seq data. Also the people working on the sequencing of this T2T genome assembly, and the cost action for giving me the opportunity to do this STSM. And that's it.
Yeah, that's it.
Yeah. Thanks for having me here. Do you have any question? Yes.
Just in the human, let's say, mammalian genomes, I've read several papers about pseudogenes that instead they look translated partially and so on. So I was interested, what is your approach? Clearly, long non-coding RNAs, but pseudogenes coding for proteins, actually, detecting proteins. And the second comment is that for the annotation, do you use AlphaFold to understand something about the protein or?
Probably the ones that are being translated, they're going to be in final annotation, maybe because of the evidence we have in biometric data. And probably the ones that are not being translated will be harder to annotate them. But maybe also Liftoff is able to find them based on the structure of those gene models in repetitive families. Maybe Liftoff is able to find them to find genes that are in extra copies in the genome. For protein data, we are only using elements of different proteins for different for other species, but not actually folding them in any way.
So I just want to say, great job, and also compliment you on working with international partners to bring the best product to the community. And that's not always easy to do. And I know that they're working on it. So I think you guys should be really commended. One of the things I want to point out is there are several groups within animals and plants that are having these discussions on nomenclature. And I really think that you guys should engage with them as well before you finally make the decision on that. Because one of the discussions are going to be, can we as plants use the same sort of recommendations, so that way if there's software developed to QC the files and things, or make recommendations for what would happen, what would be the nomenclature used for the next succession, that it might be possible to share those tools with other species.
Into a more gene name oriented, I mean, for example, in human or other species, or of other species, they are more oriented into gene models, so the gene names or gene symbols. And we shouldn't think of the gene IDs as a way to remember, a way to unique for each gene, for that more unique to a proper to a specific gene models, and not the gene by itself.
Proponent of following the things that vertebrates do, I'm going to articulate that the vertebrate genomes are much more static than the plant genomes are. Strategies they've used may not actually work well for your groups.
Okay, see. So just a follow up to the Rin's comment. I think your proposed gene IDs, for example, are not translatable directly to different species. So we need to talk also within the community, I think. Yeah.
puedes tomar el... So I had a question related to the quality of the T2T assembly and related to the length of the assembly that is 494 megabases. So do you think that this is the real size of the haploid Grapevine genome, or there can still something to polish in this version of the assembly?
Locate mostly in centromeres. I don't really know. I don't know what to trust. If it's what's the most reliable, like the way the DNA, quantity of DNA is counted. I mean, is weight, or the assembly. I don't really know.
For instance, have you found the heats, is the heat that is increased on replay in the centromere?
No, it have in one centromere, the chromosome 3. There was a drop in the mapping depth, but for the other chromosome, no, we didn't identify. Yeah, but it's highly repeated, so yeah, it's hard to tell.
¿Se escucha? ¿Se escucha?
Just a curiosity for Camille. So you did a sort of analysis of the centromeric and telomeric regions, and we tried to do something similar before in the V4 version, and then in the T2T in the V4 version was in the T2T was nice. But yeah, we saw that something weird is happening on chromosome 6, because we found a sort of telomeric repeats in a centromeric or in the middle of the chromosomes. And I think it was also clear in part from the slide that you showed. I was wondering if you noticed that, and if you have, I don't know, any hypothesis for this region, for this.
As you've seen, we are mostly focused on genes. So these are the regions that I haven't looked very carefully. But yeah, I definitely agree with you that now the centromere and telomeres are better in the T2T than it used to be in the V4. But I didn't look very carefully in these regions. Sorry. I have questions online.
We have a couple of questions from the chat.
Which are hitting this kind of quality assembly. Of course, it's easier with PN 4024 because it's highly homozygous, but with highly heterozygous genotypes, it works pretty well. So yes, and it's now cheap and very fast to assemble any grapevine variety that for sure we will very soon end up with dealing with hundreds of T2T assembled genomes for sure.
And also there was a question regarding how many new genes do we find in the centromeric regions, the T2T genome?
The answer is we need to wait for the annotation pipeline to run, and then we'll get back to this question. Yes.
Can I ask something here?
I guess this is more about the next phase of where we're going. How many are we here? 30 people, maybe 55. Yeah. How many people work on grapes? Thousands, maybe. We need how we make all this accessible to everyone and make sure that it's respected in a way that maybe the Arabidopsis community has achieved with their guidelines. Because I still see papers using V0 or RefSeq or whatever annotation they find first as soon as they Google what is on grapes. And I think we can keep meeting between us and be better, but 99% outside.
Yes.
I think here it's focused on outreach activities that can reach the limits of our community.
Continuing with that idea in mind, I think that it's a nice opportunity for us to outreach the things that we are learning here, but maybe it is a good opportunity to start thinking in Grapedia, maybe being this platform for the outreach itself. Hopefully we can do more meetings like this one, and next time we involve more part of the community, people that are more abroad. Whether we do a completely online version of the meeting or somehow an hybrid version as this one, but we should definitely think in outreach. We need to re-educate basically, and also maybe talk with somehow with the tutorials and databases, and well, hopefully Grapedia will serve as a database and will be able to somehow not obligate, but somehow give standard versions of how we should reference things, right?
But I think also that the cost action was a great opportunity to reach scientists, and hopefully some of them will stay in the Grapevine community and they would know that and keep using the most up-to-date tools or something. So I think yes, the cost was a good way to I don't like the word, but to educate the young generation in how to proceed, because we all know that it's mostly students, PhD students who are really having their hands on.
About to launch, we're about to launch like the version of this, of the version of Grapedia. And if you really want people to adopt the next version, the things that you've highlighted in this meeting where you have the workflows working like the RNA-seq, and your other one, the one that you've already put up for the network, I would highly encourage that you make sure you have that data loaded on the V5 as soon as possible, because if you don't, it just it increases the barrier for people to use that new version, and is starting as something that you're going to go out with a release right now. You want to kind of think about really setting the foundation of what you're going out with release.
Yeah. Everything on the T2T assembly and annotation right now. So it's a huge work to transfer everything for the previous version to this one. But yeah. Yeah. Really, we want the T2T to be the easiest for this, because we think this is the best that we can provide community with. So as a reference genome.
Hi, thank you. I wanted to tell why I'm using all the versions, because it's difficult to convert the positions of markers in different versions. For instance, with SNP lift, I lost 10% of the markers. And I'm working with ancient DNA, so I don't have 10K markers in my samples. So if I'm losing more markers because the conversion tool is not okay, I'm losing my time.
So transferring at least conversion tools for genes, but we are also having in mind converting any positions from any version to any other that you'd like. Because I've got my mailbox full of these requests, and I'm the tool converting any position to any.
Conversion tools
Yeah, I mentioned.
Yes, that's not before I ask for that also.
Yeah.
They don't have
Not alone.
A good reason. Yes.
We hear you.
Thank you.
And the idea would also to use this tool to convert to transfer your marker position to any not only PN versions, but any. So you can choose the reference you'd like. Thanks very much.
Thanks, Camila and Antonio. We are now going into an online. This time is Lakshay Anand from University of Kentucky. He's a PhD student of Carlos Lopez. He's gonna talk about mission he did in M together with Marco Moreto, talking about the implementation of a tool he developed that is called Chrome, that I think it's pretty nice tool for visualization and chromosomic regions. I don't know if it's already there. Hello, Lakshay, how are you doing? Let me say that Lakshay was funded through assistance of Galu Winery. So we thank Galu for allowing Lakshay to travel to FEM for 3 weeks and work in the implementation of ChromaMap. Hello, Lakshay, how are you doing?
Hi, I'm good. Thank you. How are you?
Good. We are pretty much okay here. I think so. So I leave the stage to you, and thanks.
Thank you. So thank you so much, Thomas, for the introduction. I was gonna do it myself. So as like you say, I'm a PhD student. I'm in my last year, and I'm about to hopefully graduating this. Well, yes, I'm scheduled to be graduating by the end of this year. And today I'll be talking about the tool that I developed, which is part of my thesis, and like how the change, like the modifications to this tool, which is to be integrated in Grapedia, that I did with Marcos and in the Grapedia training school a couple of months ago. So I think let's get started. So the Chroma map as a journey, it all started in 2018, which is a year before my PhD, and this is something. I would say, I mean, it's quite like personal in a way that I was just graduated from the master's program, and I was waiting for 2 months before I had to join a company, and I was basically doing nothing. So, an idea came to me that, like, why not to develop a tool? And actually, it's not more like that. So, I'm sorry, I first introduce a little bit more about me. Then, my background is computer science. I did my bachelor's in computer science engineering and my master's in bioinformatics. So I am, you can say, not a biologist, but I have always been interested in biology. Um, and grapevine specifically, in plants or grapevine, I was associated because of my association with Dr. Lopez in University of Kentucky. I do, love grapes in a way that, wine. I never worked in a vineyard or anything like that, but I work with data. I work with other species too. So I'm sorry for, getting off track. So coming back to this journey of how, like, then I joined this PhD program, and thankfully, Dr. Lopez agreed on making this a part of my thesis as well and bringing about more and more changes. So over the period of time, like, as you could see that I had released major versions of it, and except for in 2020, because 2020 was like horrible, and until 2022, when I aim, especially with Dr. Lopez specifically asked me not to work more on it, and or to I lose the inspiration to do it as well, like for one, up until, as you'll see later, um that how it's going to continue. So let's start with what ChromaMap actually is. So a ChromaMap is an R package. It's a graphics package, which is written in, of course, R, JavaScript and the background, and D3, which is a JavaScript library. So it's a genome visualization tool which allows you to visualize genomes, chromosomes, or genes, and to map genomic features such as genes or SNPs onto those regions. So one thing is that it allows you to create interactive plots. So if you have ever used like Plotly or plots, which when you hover over it, it kind of like more interactive in nature. So it gives you like more information or something. And on top of that, so you can also visualize numeric or categorical data associated with your um, annotations. So such as gene expression, epigenetic modifications, you name it. So it works like a chromosome is being visualized as a contiguous genomic window. Each genomic window, of course, can have more than one annotations. And when you hover over it, a tooltip kind of shows more information about the genomic windows, like how many uh, like how many annotations are there and so on. So in terms of how it works, it's pretty simple in a way that you create input files, which are tab delimited files that's similar to like bed files, and or it can optionally also use R objects if you're using it in R, and you call it chroma map function, and voila, you just have the plot. Oh, sorry. So the files are pretty simple. You provide a chromosome file with the information about the chromosome, the region, the start and stop coordinate, and optionally the centromere. It also visualizes the centromere. And the annotation file, which of course have the genomic information of the annotations on it and the associated data. So this is like a simple chroma map where you can see the chromosome, and as you hover over it, it's showing tooltip where telling the genomic range, the number of annotations, and each of the names or IDs of the annotations that you had passed. So two types of annotations is possible in ChromaMap. One is the point annotation. So when chromosomes are considered very large in size, so most of these annotations will be very small, maybe more like accumulating a lot of them in a genomic window. So a point annotation kind of visualize it as just one genomic window. Or in opposed to that, there's segment annotation also, which allows you to visualize like genomic features. So one of the feature which is like a multi-track or a polyploidy feature. It is called a polyploidy feature because when I developed it, it was more like polyploidy in mind, like to be able to add chromosome sets. So it literally works that way. So you pass sets of chromosomes, and these chromosomes set can have different like number of chromosomes as well. And of course, you could see the different lengths of the chromosome. So since there is no restriction on the number of chromosome and the length of the chromosome, this polyploidy feature can also be used as a multi-track. And so basically, with those features, there are many applications in general for ChromeMap, and which could be applied when included in Grapedia, like for comparative genomics, like comparing the different versions of the Pinot Noir assembly. We can compare different cultivars, like different sample types or tissues. It allows layering of data, and you can compare the samples as well. So now a little bit deeper into like the data visualization of the data that are associated with those annotation. One way you can do it is through heat maps. So heat maps are basically use the color to visualize the numeric data, like for example gene expression and differential gene expression or log FC or something like that. And then on hover, the tooltips shows the heat map for the annotated genes or features within that genomic window. So when coming to the numeric, so for the genomic window, when you hover over it, it shows the heat map for individual features, but the aggregation, so like the summary on the like genomic window can be summarized with different functions like the average, the sum, minimum, max, or even count when you want to show the density. So the heat map for the continuous data or the numeric data, and there's also option for the discrete data, the discrete color maps. So in addition to using the colors heat maps, you can have like the bar and scatter plots of the numeric data. And so epi tags is, like a feature that allows you to put a tag on a specific location or something. This could be useful, for example, to visualize in the methylation, for example. So one specific feature about having filters on the data. So like as you can see the scatter plot and the bar plots, and you can apply these mathematical filters to filter colors, for example, differentiating between the positive and negative values, and so on. And using it within the chroma map is also easy. So this is how you pass the label and the mathematical operators on it. A multi-factor scatter plot can also be used. So this is like a scatter plot, and on top of a scatter plot, another dimension or the factor is used, which is a categorical one. So like two information can be visualized at the same time. So this is an example of using the real data for for the transcriptome data of the COVID patients, and it shows like in different layers, the gene density, the log FC, the fold change of the expressed genes, and yeah. So another feature, major feature is a Chrome links, which is having these Bezier curves relation links between two annotations. And what's interesting about it is these links can be made not just between the chromosome of the same ploidy, also chromosome from another ploidy or another way, another track. So it could be useful for for the visualization of like orthologous chromosomes or orthologous genes, I'm sorry, on different species or cultivars. Additionally, on these links, you can associate a continuous or a discrete data visualized as colors. So this is just a syntax of how you can simply form a link between the two annotation with two columns for first annotation, for second annotation, and the associated data. So this is more like chords are for the segment features, and it's visualized the segment features. It can also be colored based on the discrete or continuous data. You can also add edges to it to show the direction of the relationship or directional relationship. So this is an example of comparing the orthologous genes between the mouse and rat genome. So another feature that I recently added was like a 2D chromosome visualization. So the way it works is you have the genomic window, and on the y-axis are the annotations itself being spread out. So the only thing is that the y-axis will showing the window up to the window length. So this is one example. So this could be useful to know how much distributed or dense your region are, because this is literally showing annotation locations, because the genome size is large, within a genomic window, there could be many features. So this spreads out those features to show kind of like a density or location map. And in addition to that, a secondary information like continuous or discrete information can be visualized as colors. So the maps can be made with high resolution. You can do adjust the window scaling size by factor, or you can also have the fixed window kind of thing. So additionally, there are some more features. These are, simply like annotation labeling, highlighting with grid lines to highlight a specific region. In our version of the chroma map, there's a zoom in feature where you can pass this argument to zoom in on a specific region. Hyperlinks. So one thing is, while inputting the data, you can add an hyperlink to each of these annotations. So on an interactive version, when the user sees this tooltip, they can select these annotations and could be landed to any of the standard database. So it's an extensive graphics package that allows a lot of customizability, from little details of adding title, the size, the colors of annotation colors, and so on. And there are inbuilt export options, which you can export the plot as PNG in SVG. So this brings us to since it's an interactive plot, so you can export it as a static image to be included in publications, but you can also use the interactive version as a standalone HTML document. Additionally, it can be used in R markdowns if used in R in Shiny web applications, because it's an R package. It's easy to integrate into Shiny applications. But so far, for the web applications which are not Shiny based, it cannot be easily used, at least until now. As I'll discuss that later. So far, the availability of Chroma Map the stable version of it is available on CRAN, like other R packages. There is something that I developed as a Shiny app for it, which is kind of more experimental than, but it's available as a containerized web app. I also this to disseminate this a platform where you can know more about Chroma Map and so on. It has been published. It was finally published last year with few citations. This is some of the examples people have used it. And I have to say that as for this 5-ish years of the journey of releasing each and every update, there has been a great contribution of the users, of the people, of the researcher who have been constantly contacting me with their very positive feedback and asking for if they could do something with Chrome and then give me the not just the inspiration, but also motivation to be able to implement those features one such interaction I have to say was happened in the Grapedia training school with a lot of very nice discussion, which resulted in, which kind of like also now motivating me to add more features to it. So, like I said, so that Grapedia Training School marks kind of like another chapter for ChromaMap. Its integration with Grapedia and its transformation into another tool is quite an exciting thing for me. And have to first acknowledge Thomas, Marco, and Marie to be able to give that opportunity to me. It was an incredible time. It's been 2 months, but it has really been very good time for me. I mean, very yeah. So like and of course I would like to thank the funding agents, Gallo Winery, and the Cost Action. So now a little bit about what are the changes needed to be done and which has been done for the Chrome map to be able to integrate in Grapedia. As you have already seen, Grapedia is not a shiny web app, and it is developed basically using the React JavaScript framework. And so one of the thing that what our the Chrome app lacks so far is that it's been written with R and the JavaScript. The backend, the actual visualization part is JavaScript and as well. But I wrote the R, which was doing the I would say quite a bit of a part for the processing of data, the annotation, and so so one of the proposed thing, and which has been accomplished when I was in Italy, to be able to fundamentally change the entire thing into a JavaScript. So as a part of a Grapedia or in general, Chrome app is now or will be available as a Chrome app JavaScript component. So which can be used as any other React component, like how the Cytoscape was used for network visualization or other things. So it can be easily integrated into Grapedia. And another thing, by making this completely like JavaScript based, has opened up opportunities to include features which wasn't totally possible before, having more control over it. I'm going to go through some of the features that I have implemented already to say that how it's gonna look like. So one of the things it's going to have, and these features will be released with the release of the JavaScript version. So one of the thing is having a toolbar. So there will be a dedicated toolbar every time the user plots, or it will be like available to the user where, there will be the navigation for the buttons. Properties will be for where, users can adjust properties and so on. There will be there's now a zoom feature which is integrated into the platform. So like the user can zoom and using the toolbox itself, because the JavaScript object is storing the whole data. It was possible now to have this kind of zoom feature. So in addition to that, there's a context menu. So while aesthetics of the plots are usually not considered important, I always think that it's kind of I wouldn't say, of course, it does not have any significance biologically because the visualization serve a purpose of showing something. And then the aesthetics, the color, and those things should not be that difficult for the user to change something. So these context menus now allow to change the aesthetics in a very simple way. And an additional feature like is not really I mean, which was possible with JavaScript is the draggability. So these components can now be dragged in place. Every component in a plot is now draggable. So it doesn't have to be necessarily done programmatically. The user can, rearrange the components of the plot. So yeah, these are some of the major features that I have successfully incorporated, and hopefully more will come when the final release of the Chrome app will be done along with the Grapedia. And yeah, and it will be released as I said, a JavaScript library, and maybe also an R package or a Node.js module, which will be easier for its integration in Grapedia. And so, yeah, that's pretty much it. Thank you. I cannot hear.
Next question. Do you hear that, Jay?
Yes, I can have.
Hi, thank you for the nice presentation. Very impressive. I have a question regarding the export of data. Can you understand me? Yes. I have a question regarding the export of data.
Yes.
So when you zoom in, you say you can export it via HTML and other formats, but is there something like a simple lookup table where people can easily work with? They only focus in a candidate gene region, or yeah, like a window Dario mentioned between flanking markers, all the features listed.
Yeah, so it is something that I have considered, and it's also something that came out of a discussion in training that a lot of people wanted. The export of the data as well. So now that the data is in the JavaScript object form, I will include the export of the data in view. So that way, into like more like a table or a CSV TSV format in which whatever the user is looking can export the data.
Thanks, Lakshay. Any more questions? Marco?
Thanks. Hey, hi, Lakshay. Nice to see you again.
Hi, Mark. Yeah, nice to
And
meet you.
thanks for the nice presentation. I saw a lot of new things since last time we chatted, and I know I probably shouldn't ask this question, considering your duties with the PhD defense and everything. But I don't know if you are if you have more or less an idea about when the JavaScript version will be ready, let's say, for be used in production, let's say.
I will give. I'm trying to like wrap up the PhD thing. Fortunately, I will be able to use possibly as an chapter in the thesis also. So like it will be like not an extra project for me. So I will try to release as soon as possible, especially now it has been officially on Grapedia. So I will do my best to release as soon as possible.
Yeah, I don't want you to. I don't want to force you to tell a date because probably your boss is listening. So no pressure at all. But yeah, it was a really amazing job. It was really nice working with you, and I'm really keen actually to see ChromoMap implemented for all the tools that we already have in mind. So thanks a lot.
Thank you.
I hope that it was as good as it was for us to have you in the training school and during your stay in Fed. I hope we have also great you felt part of this meeting. Thanks once again.
Okay, so welcome everyone to this dashboard tutorial. So this is I'm going to introduce you to the web portal as it is now. First thing that I have to tell you is that probably what the prototype will have, it will be similar to this, but maybe some changes will be or some functionalities will be added. So what you are seeing in the screen right now is what you will see the first time that you try to connect to the web portal. As you see, we are in the login page, and if you don't have a user in an account, you can always click on the register button, and you will be taken to the sign up page where you can introduce your name, your mail, and your password, and take into account that the email has to be an institutional one. After introducing all the data, you will receive a mail to confirm the creation of your account. When you have registered, you can go back to the login page and just enter your user and your account that you just created and click in the login button. When you do that, you will be redirected to the landing page that contains, as you can see, a brief summary of what is the Grapedia initiative. On the left, you have several tabs as profile. That is the one that you are seeing now, where you will see the user that you just created, and also the list of analysis that you have done till that moment. Also, you will have the datasets tab, where you will see a list of different datasets. There are all of them are fully downloadable just by clicking on any of them. You will be redirected to a site like the one that you are seeing with different method information and a download button. It's important to mention that the metadata will be, of course, of high quality, and this is our way to ensure that all the data that is uploaded to the platform is free data. Then we also have the workflows tab, where you will have different Nextflow workflows that you will be able to download and run in local in your PC. Eventually, you will also be able to run it in the Wikipedia portal using the Sequentia tools. Then we also have the tools tab, where you can see that now we have a functional BLAST and a functional gBrowse2. If you click into the BLAST tool, you can introduce any FASTA sequence of your interest, select the type of BLAST that you want to perform, and also select the database you want to do the BLAST against. Right now, we only have these two databases, but more databases will be added in the near future for the release of the prototype. If you click on the gBrowse2 tool, you will see a fully functional gBrowse2 where you have different tracks that you can select and navigate. Again, for the release of the prototype, we will add more tracks. Then you have the search tab, where you can introduce the gene ID or the gene name of any gene of your interest. For this example, MI15. Once you click the search button, you will retrieve all the genes that have the string that you have looked for in the gene symbol. In this case, we have to scroll down until we find our gene of interest, MI15. Once we found it, you can see that we have 4 different buttons: gene card, gBrowse2, BLAST, and UniProt. I will show you the gene cards button. The BLAST button takes you to the BLAST of the gene, allows you to do a BLAST of that sequence. The UniProt takes you to the UniProt of this gene, and if you click the gBrowse2, it opens the gBrowse on the gene of your interest where you can check the differences if there is any difference between the different versions of annotations. So B4, B2, B1, and B0. Again, new tracks will be added, so all the tracks will be available for the users if they do what I'm showing here. And finally, we have the dashboards tab, where right now we have three functional dashboards: the gene cards, the multiple gene, and the networks dashboard. All these dashboards are based on React, and well, I'm going to show you the example of the gene cards dashboard using Me15. You can introduce a gene of your interest using the gene name, the B1 or the B3 gene IDs. Once you introduce your gene of interest, the dashboard will autocomplete your search, and you can click the search button. When you do that, you will see here on the left all the information available for the gene that you look for coming from the gene catalog. We have added some useful links, such as, for instance, the Grameen link that can take you to the Grameen page of the gene that you are looking for. Thanks, Doreen. Eventually, we will also add the same information regarding the Cabernet Sauvignon orthologs, and also the orthologs for all the cultivars that we may add into this Grapedia platform. On the right, what you are seeing now is the box plot, exactly the same information that David showed this morning when he showed the gene cards app from the Bitviz platform. As you can see, it's a fully interactive box plot where you can click in any link, in any dot that you want. Then if you scroll down, you will see the DAPSEC data. In this case, we don't have any DAPSEC data because what is contained here is the peaks detected in the promoter region of the gene that you have looked for. In this case, we don't have any DAPSEC data that is binding to the MIP15 promoter. And below the DAPSEC data, we have the gene co-expression network, meaning the list of 420 genes that are most co-expressed with the gene that you are looking for. As you can see here, we are finding some still benzene tases, such as the 31. And if we select another network, such as the tissue independent, you can see again that we still have some still benzene tases co-expressed with Me15. So now we may want to look for the still benzene tase in the gene cards dashboard and check what we can see in this gene. So again, when we click the search button, we will retrieve all the gene cards, all the information present in the gene catalog for this gene, for the still benzene tase. And we can also go to the Grameen link, but in this case, this gene is not present yet. This is important to mention that the Grameen links are generated on the fly, and we are not checking if the Grameen link is functional or not. But eventually, if some information is there, the link will be working. So here for the still benzene tase 31, we also have the box plot, and now you can see that we can see DAPSEC data. In this case, we see that Me15 and Me14 are binding the promoter region of this still benzene tase. If we scroll down again, we can check the co-expression table of the still benzene tase 31, and you can see that there are a lot of still benzene tases co-expressing with the still benzene tase 31. So now that we know this information, we may want to check if there is visual co-expression. If we can generate a heat map with the expression of these genes of Me14, Me15, and some still benzene tases across all the runs that we have used for creating the networks that I showed this morning. For doing so, you can go to the multiple genes dashboard, when you can introduce one by one the genes of your interest in the search tab, as you are seeing right now. So I'm introducing Me14, Me15, and I will introduce also some still benzene tases. If you click the search button, you will generate automatically an interactive heat map where you can move your mouse around and check which SRA study is the expression value coming from. And also you can select the tissues that you want to show in the interactive heat map. If you scroll down, you see that the dashboard also generates a conversion table where we show the B1 ID, the B3 ID, and also the gene name of the genes that have been searched. However, since it may be a little bit, well, it may take you some time to introduce a lot of genes if you want to analyze more than, for instance, I don't know, 10, 20 genes at the same time. We also generated the option of creating an Excel file like the one that you are going to see now, like the one that I'm showing you, where in the first column, you can add the gene name or the B1 or the B3 IDs of the genes you are interested in. The rest of the columns are not going to be taken into account for the generation of the heat map. So you can add here as many information as you want, but you have to take into account that only the first column will be taken as an input for the dashboard. When you have created the Excel file, you can just drag the Excel file, as you're going to see right now, to the browser tab. And the genes will be automatically introduced into the search into the browser. Again, you can click the search button and you will have a new heat map with all the genes that are present in the first column of the Excel file. Again, the heat map is fully interactive, and here you can see the conversions table with all the genes that you have seen in the Excel. So now what we can do is go into the networks dashboard and check if there is if the networks detect co-expression between all the genes that we are seeing in the heat maps. Again, here, this dashboard works exactly in the same way as the multiple gene dashboard. You can just drag the Excel file, or you can also click on the browser to look for the Excel file that you want to introduce manually. Once you select it and you click the search button, the dashboard will query the backend to retrieve all the co-expressions between the genes that we have added in the browser. Then the Cytoscape component will generate these networks using the Cytoscape JavaScript library, where you can see that there is co-expression between all the genes that are present in the Excel file. Again, the networks are interactive, meaning that you can drag and move all the nodes of networks as you want. As you can see, if you scroll down, you will see that we have also implemented the one gene network of Stefania that she will be showing tomorrow. Again, these networks are also interactive, and you can change the relative frequency threshold in order to make your network more stringent or less stringent. So yeah. So we know now that these genes are co-expressed, but we may want to check if there is co-expression between these genes that are coming from the stilbene pathway, the regulators, and also we may want to use the networks as a clusterizing system, as I showed you this morning. For doing so, we can generate another Excel file, like the one that I'm showing here, where you can see that we have, again, the stilbene synthesis, M14 and M15, but we have added more genes that are coming from different secondary metabolism pathways. Again, as I told you this morning, although we are using this tool for clusterizing genes for different secondary metabolic pathways, you can still use it for analyzing genes coming from different pathways, such as, for instance, I don't know, involving different functions, such as, for instance, resistance to biotic stresses, abiotic stresses, or development, or whatever you want. So what we're going to do basically is uploading this Excel file to the dashboard and see the networks that are generated. What we are going to do is to refresh the dashboard and select the new Excel file with all the genes that I showed. Here you can see that the networks are actually clusterizing the different genes according to their co-expression, and you can clearly see that we are able to separate the genes coming from the stilbene pathway and the ones that are coming from the anthocyanin and proanthocyaninase pathway, meaning that the networks also here in this dashboard good clusterizing tool. And this is everything that I had to show you in this brief tutorial. I would like to say that a new tutorial, maybe more complete and with all the functionalities, will be also uploaded to YouTube once the prototype is released, and it will be available for all of you in YouTube. So as always, thank you very much for your attention. And again, we're open to hear all the feedback of the community, since this is a portal that is made by the community to the community. So thank you very much for your attention. I don't know if you have any question. Yeah.
Thank you. Congratulations for the huge work. Very nice. And I was still thinking about gene expression networks and sick data. I don't think it's easy to go back to the when you select. When look for the transcription factor associated to cell by synthase genes. Oh. Yeah, when you show that the trick, look 14, 15, something else appear. Next one. Okay, here we go. I was wondering, so an additional layer of validation for the DAPSIC data, as you know, is the fact that they are also correlated with the genes. So to identify high confidence target, you can check whether the three transcription factors are identified by DAPSIC and then look at the gene expression. But of course, there you have all 420 genes that are correlated with the genes. So perhaps it could be easier to, I don't know, to add an additional column in the upper part where you indicate whether the MIB 14, 15, or whatever are also co-expressed. So if you have this additional validation, I don't know.
Absolutely. Actually, it's a really good idea. It's actually really simple to do it. So definitely it's something that is doable and really quick to see to do it. So yeah, absolutely. It will be added. It's a really good idea. Yeah. Yeah, absolutely. Yeah. I would also like to say regarding the DAPSEC data, that the idea would be to use the chroma map package that Laxey just showed, in order also to show the peaks of the peaks and the intensity of the binding. For instance, in this case, the 2 transcription factor in the promoter region of this stilbene synthase. So I think it could be also interesting to show it in a more visual way. Sorry. Wait a second.
Okay, here we are. You know that I was playing a lot with the dashboards these years. So my suggestion in some cases, for example, with the XR app, it might be really important to have the clustering working in a proper way, and also the case of the name of the samples that are the one that have been uploaded, so there are codes. So if you are exploring a data set, the names be easily understood. So just choose a class of the tissue and that kind of things. So because when you visualize an heat map and you are exploring a data set that you don't know, the codes are just meaningless to you. So it's really, really important to have that showing up very easily.
Yeah, absolutely. Yeah, absolutely. That's definitely a good idea, and it will be taken into account for that.
Sorry, yeah, a really good idea. Something that you usually do with the X index, like with the data frame, you have like a multi-index that helps you out in clustering your samples, considering like one extra layer that might be the tissue or specific conditions. But the problem is that I mean, we rely on external libraries for plotting the things, and it's not always something that can be done directly. So we need to write extra code for doing that. And because like I'm thinking like in Pandas is something that you visualize really easily in Seaborn and with R as well. But with JavaScript is not like that straightforward. But yeah, anyway, it's a really good point anyway. Thanks.
Thank you, Luis. Very cool. Really amazing work. And I'm actually happy to see the CAB orthologs that you have in the gene cards. And I wonder if those are links or just listed IDs so far.
So right now, they are just text. They are it's actually the orthologs that we have in the reference catalog when it was published. But definitely, so it's something that it would be really good to talk about how to link how to make this a clickable link to take you to Grapevine Genomics or the key browse of your Grapevine Genomics or any other website that we want to link the information to, because it would it will be easier for the users to jump from one side to the other. And in the future, for sure, there will be clickable links. Yeah, that's the idea. Sorry. Thanks.
Press and see it actually working. So if you thought about this goes to clustering of sometimes when you have 9,000 data sets, it can be really overwhelming. If maybe you might want, since you've done the clustering already, you might want to come up with a slim set that would, where you have data sets that are really redundant there, to make it more usable, at least initially when people are looking at the data sets, because it's, so I'm just saying this because it's amazing. And as people are adding more and more of those sets, the slicing of those to make them more usable are probably going to be important to your downstream users. And so I'm not sure if you want to start out with having like a reduced set versus everything.
The problem for the visualization. Well, one way to solve that it's not to have JavaScript doing the visualization and have it like a static image. Yeah, I know it's not. Yeah, the problem is that you sort of lose. The problem is that you sort of lose the look and feel that we have, like consistency. But yeah, and on the other hand, working with let's say more than I would say 10,000 points also for networks and visualization. I mean, we can simply dump the responsibility on the client side, like say you should buy a laptop with more RAM and just ignore that. And but yeah, I know I totally understand the problem.
I also have another question. You said initially you need an institutional email address. Was there a reason why you guys made a decision on that? Because there are some institutions, like in the US, that don't even have an institutional email address. They're using email for their regular accounts. Do you see that being the limit?
Thanks. I see you're enjoying your role. Yeah, actually, that it's a standard policy from what we started developing since we started with the template that were given us from Sequentia. And I mean, I totally understand why they want, real email address or at least something that is linked. But I mean, it's something that can be changed and adjusted. And of course, it's just to avoid hacker.
Yeah. But also like we can, let's say, simply implement other ways of having, signing in. So from other links. So if you already have like another account in other platforms like Google, and I mean, it will totally work.
Marco, on the same subject, so can we use like some production does when you are like invited? So let's say all the people that have been in, but we do have the email in what we participated to the meeting, and the one that we have an email list, and each of them like will have 10 invites and do things like that to avoid.
Yeah, totally. Yeah, absolutely. It totally makes sense. I mean, we are talking about a community, the Grapevine community, that is not that large. So, I mean, I won't say that we know each other completely. So it's not a fully connected network, but still, I mean, we're not talking about millions of people. So this is absolutely something that is doable. So to go through invites, yes.
Well, if no one else from here wants to ask something, I just saw that in the meeting there is a question that they are asking if co-expression data is limited to the gene of the genotype displayed, or it will include expression in orthologs from other genotypes. As it is right now, all the expression is coming from alignments performed against the PN424 reference genome. So right now it's limited to this genotype. Obviously, in the future, the idea is also to do it against other reference genomes, and therefore you can check the you will be able to play around also with expression in orthologs with from other genotypes. Yeah. Yeah. Yeah, I know. Yeah. That is the point, yeah. So in the future.
No, but it's not limited because you are seeing
Okay, yeah.
No,
Yeah.
We could separate them, yes.
Yeah, but something that we haven't done yet. Sorry, wait a second.
I'm talking about, of course, the co-expression. There is a work that I didn't do, but it's based on what I did with Vespucci, and Vespucci is based on the V1 on the reference genome. So the problem is that even though you have experiments coming from different cultivars, you lose that information since, like, if you don't have the gene, as Dario knows, I mean, there's no way you're going to align against something that is not there, of course, but this is the trade-off. So in the sense that if you want to think to be comparable, you have, to have like the minimum that is common to everyone, and you're going to compare something that is in common. And I know it's not ideal, but the alternative will be, sorry, no, don't worry. And, but the alternative, it's, this is the next micro, Anyway, the alternative is actually to have different transcriptomes and performing the alignment on, let's say, Sauvignon or Cabernet, so for each of the transcriptome, and then have sort of orthologous maps from one transcriptome to another one. But again, you're going to lose the information since if you want to see what is in common, you're just going to see what is in common and not what is unique. So again, it's like a trade-off. But the information you might have, it's the expression related to the V1, V2, or V5, and then the possibility to jump from that ID to any other ID for any other species. But yeah, that is the maximum you can get. We need the pan transcriptome.
Thank you. Thank you, Luis, for really the huge amount of work and very nice
this dash representation of the data, it was a surprise to find one gene that we were really happy to see. And no, it's just a comment that maybe we will have time to discuss in this meeting on how to present. And because we are producing other releases, so maybe we will discuss if make a very long page or doing a different, let's say, tab for association network, also to differentiate, because also we will have regulatory networks with oriented edges that probably have a different meaning. So maybe we can do a dedicated one to co-expression, the other to causal networks, with also some explanation on the meaning of the edges and colors and anything we can add. So thank you very much.
Yeah, you're welcome, of course. We are always open to discuss the way of showing the data, and if you have more data, of course, yeah, we can. If you prefer to have a different tab or a different dashboard, it's never going to be a problem. So yeah.
Okay, I think that we are ending the day of this annual meeting, the first day. I think that we have learned a lot of how the Grapedia portal has been implemented during this first year of the SIG grant. And the next days, we will have a lot of talks about the new advances in Grapevine computational biology, methods, and many things. But now we are finishing the talk with Ana Conesa. Ana doesn't need really introduction. I'm pretty sure that many of you know her. And I'll just briefly introduce her background. So Ana actually is now a research professor in the Institute of Integrative Systems Biology, where am I also. And she is a senior scientist in informatics and computational biology. She's also a merit professor in bioinformatics at the University of Florida, where she was before coming to Spain. And she leads here the Genomics of Gene Expression Lab, as you will see. Ana has built a scientific program focused on the development of algorithms, and I'm pretty sure that many of you have used packages, algorithms, or software that she has developed, such as Blast2Go, Masic Pro, Noisec, Quantomics, and many more. Today, Ana will talk about using long-read platforms, sequencing platforms, for unraveling annotation issues, right, of genomes. And the stage is yours. Thank you very much for being present today with us.
Well, thank you, Thomas, and thank you everybody for being here and listening to the last talk of the day. So my title is here. It's talking about long reads and insights on the complexity, the functional complexity of the transcriptomes. I have changed a little bit the orientation of my talk to talk more about identification of transcripts and annotation, but I will say a few things about functionality as well. Okay, so you probably know that long read sequencing was the method of the year by Nature Methods this year, and this is because the technologies like PacBio and Nanopore now are generating very long reads. And for the analysis of the transcriptome, that is what I do. The advantages of having long reads is that you can resolve the alternative isoform structures. These are difficult to identify with short reads because they don't have contiguity across the exons, but with long reads, in principle, you can obtain the full-length transcripts in one go. And why we want to know more about isoform, because, they are claimed to be involved in, different processes. Different isoforms may have different functional properties, like having different binding affinities, of changing the localization of the protein, and therefore also the function. And also different isoforms may have different binding sites for regulatory elements like microRNAs of RNA binding proteins. So they might be a differential functionality associated to them.
Also, names have been used a lot to annotate transcriptomes, and what we are seeing these days is that there is an explosion of papers reporting novel transcript, novel isoforms found by these technologies, even in very well annotated organisms. So here you have a paper last year, nanopore profiling transcriptome in the GTEx, so the human tissues database, reporting 70,000 novel transcripts. And for example, in the plant world, we have just an example, right? There are many of these. Arabidopsis genome with hundreds of 170,000 novel transcripts found by this method. You look at the data, and this is how some human data looks like for this gene. Here, what you are seeing is the genome browser annotation of this gene in this particular cell type, so human cell type. And this data results of the compendium of different transcriptomics of long-read sequencing methods and platform and ways to analyze the data. I will talk about this in a minute. But what I want you to see is that here you have the gene code annotation for that gene, so the 5, 6 alternative isoforms being annotated for the gene, and these are the transcript models that you obtain with the long-read sequencing technologies, right? And then you see things like but there are some areas in which you have intron retentions, and then some other areas in which you have a lot of diversity. And these are final transcript models claimed by the technologies. Of course, the question is all this real? There's many through isoforms of so basically, I would say that there are a number of challenges in long-read sequencing. Of course, is the accurate code of the transcript models with the technologies. There are a number of technologies of tools, bioinformatics tools to analyze the data. But as I mentioned before, there are a lot of novel transcript being detected. And of course, we would like to then know which is the functional implication of this transcriptome complexity. So are different isoforms really doing different things, and can we predict what? So my lab has been working on this problem already for 10 years, and we have developed a number of tools. We call it the functional isotranscriptomics framework, in which we try to go from the long reads, the annotation of these transcript models using them, the functional profiling of the different transcripts, and then doing some statistical analysis and systems biology approaches to understand in the complexity of the transcriptomes that are found by the long-read sequencing technologies. So today I will be talking mostly about this part because it's related to annotation. There is a lot to say about these two, and you can read this paper in 2020 in which we are describing the annotation and the how to analyze the data. Just very briefly about what we do for the functional annotation of the transcript models. It's simply we run a number of tools and ways to predict to map annotations from databases into this diversity of transcript. Many of them are novel. Both at the RNA level and protein at the protein level. And then the annotation layers that we can have here are things like upstream open reading frames of 3' UTR and 5' UTR, cis regulatory elements, microRNA binding sites, RNA binding protein binding sites. So we can collect these and annotate the models at the RNA level, but also at the protein level. We put together things like the PFAM domains, the PT modifications, whether there are active sites, transmembrane domains, nuclear localization signals, disorder regions, all this kind of information come together, and then you can see how they vary from one isoform to the other. And then with PAPAS, which is a tool to analyze all this data, you can put it and then make questions like what is the expression of isoforms and what are the functional implications of it, right? So I will not talk more about this here now, but again, you can look at the paper and see how these tools work. And if you are interested in using them, you can contact me back, and then we can show you further how this works. But I will be talking more about the identification of the two transcript models, because I think that that's probably one of the hottest issues now. and how we can use also scanty fabric. Okay, so this is the problem, let's say. Trying to address this problem, a couple of years back, a number of people working in the field came together to set up what was the longer GAST challenge. So the challenge was to file the methods for transcript identification and quantification using long reads. So what we did basically was to generate data that was put in the public domain and ask developers of bioinformatics tools to use this data to predict test models, and then we will evaluate the results. The data that we put together was human, mouse, and the manatee. That was with the idea of having a non-model organism. Of course, human and mouse are heavily annotated, so there is a lot of information about them, and probably it's easier to do the job there. We have different cell types. We have a combination of the mixture of cell types. We also created synthetic data, so computationally generated data. And we also had in these samples, we have spiking. So these are RNAs that you put in the sample, you know what they are, so you can evaluate whether the tools are able to recapitulate them. We use 4 different library preparation methods. The most common one is that you create first the cDNA, of course, but you also have, as you know, direct RNA sequencing with nanopore. There is this methods R2C2 in which they create a kind of circularized molecule that is sequenced several times with nanopore because it's first amplified. So this is a protocol specific for nanopore. And then we also have the cap trap, or the method in which you actually capture the RNA from the 5' end, and with the you will be captured at the end of the transcript. So 4 library preparation methods, which we sequence with sequels. So with PacBio, MinION. So we had sequel2 at the time, MinION, and ILU. Just before I forget, this is the consortium that participated and that put this project together. So basically, we had a team that was the evaluation. So the evaluators were not participating in the contest. So we were not predicting, we were just evaluating the predictions made by others. And it was Fran from my lab who did most of the work that I'm going to show you here, because it was scanty3 based. And then we have other people doing experimental validation, looking at quantification, looking with gene code, manual annotation. I will show also results of that. And these are the different labs that participated, provided predictions that were evaluated. So we have 3 challenges. The one was for transcript identification with a genome that was well annotated. The second was quantification, and the third was the same, but without having the annotation. So starting with the challenge first, the challenge 1, we have here the human and the mouse data, the spikings, remember, so this synthetic RNA, also a mix, and then the computationally created data, the synthetic data. Okay, so first, maybe we can have a look at the data itself, right? You will see in my talk that I will show you like these 6 different combinations of library preparation methods and sequencing platform. Okay, so for example, this means that library cDNA and then sequence with PacBio, right? And in total, we had 6 combinations. Here you have the length of the reads, right? So not transcript models yet, just the reads. So what you can see here is that the combination of cDNA and PacBio gave overall the longest reads, right? The distribution with the longest reads. Also, R2C2 with nanopore were given long reads of longer reads. But for example, we have shorter reads with direct RNA and the combination of CapTrap with nanopore. So this is a clear difference between the technologies. Here you have also other quality parameters. This is the number, so the sequencing depth. So with cDNA and nanopore and CapTrap and nanopore, we were able to generate, let's say, with the same amount of number of reads that were in the range of 20 million. But for the other combinations, for example, direct RNA with nanopore or cDNA with PacBio, we were in the range of like 2 million. So it was a 10 times factor difference here. However, other parameters like the quality, which is here assessed by read identity, so how many mismatches and errors with respect to the reference, it was clearly better for R2C2, right? And cDNA PacBio here than for the rest. So these are the ones that also have the longest reach, right? So you see here all the differences in the quality and the characteristics of the data. We had in total 6 library preparation methods and sequencing platform, as I showed you before. So that's the magnitude of the challenge. 3 samples, right? 2 human and 1 mouse. 2 we gave to the participant 2 analysis options, so you can use just the reads of the combination with short reads. Some people were doing that. So that was in total 36 different analysis possibilities. And we had 11 different submissions, right? These are the tools, the bioinformatics tools. So there were like 3 types of them. So some of them were really heavily based on the reference annotation. And here we have Bamboo, Flames, Flare, and Isoquant. They are really using a lot of the annotations to make their predictions. Some others are kind of halfway using the annotation, halfway trying to guess from the data. This is ISO-IB, ISO-Tools, Mandalorian, Spectrum, and Talon. And then we had one that participated in this challenge, but actually was not using the annotation for predictions. This is Lyric. Okay, so if you see a little bit of the models, the predictive models, and we try to somehow indicate what is advanced related to the technology. So what you can see here is what happens with the different sequencing platform, Nanopore and PacBio. So the transcripts that were, for example, detected only by PacBio were in general longer, had more exons, and also were detected with lower counts per million. So they were on lower expression levels than the ones that were only found by Nanopore or by both technologies. So it's like a longer transcripts by PacBio. The reads were also longer, so it makes sense. If we now look at the library preparation methods, what we see is that R2C2 and cDNA, right? Exclusive transcripts were again longer and had more exons and were detected with lower levels than the rest. Okay, so what we see here is that there is a clear relationship between the quality. So remember the length and the quality of the data, and the kind of, in principle, the quality of the result, right? And also that is more important, good quality that really allows a high number of reads, right? Because those technologies that were producing 10 times more reads were not giving us the better models in principle. Okay. Okay. So that was in general how the data looked like. But as I mentioned before, we use Scanty3 to evaluate the data sets. And Scanty3 is a framework for the quality control. We do also some filtering and annotation. Actually, we use this part here of the quality control of the data with a method for the long reads. And so the Scanty does, so that this kind of became like a well accepted in the field, is what we call the Scanty category. So we annotate and we describe the long read transcript models based on how they compare to the reference transcriptome. And we call full splice matches to those transcript models that are matching a reference at all junctions, right? So in principle, this is a known gene, a known transcript. Then we have the incomplete splice matches. They're matching a reference transcript, but they are missing some exons at the beginning of the end, right? So these are like fragments, could be also alternative 3' end or 5' end, but they're missing some of the junctions at either end. Then we have what we call the noveling catalog. So basically here what we have is that these are combinations of existing donor acceptors. So that this means that these different blocks are combined in a different way. We also have the novel nodding catalog, in which we have like a new donor acceptor site, right? That is not in the reference. So this is a completely new isoform. Then we have things like intra-retention. We have also treatment transcripts, antisense, but I mean, normally these are the majority of the transcript that you detect. And then Scanty does is integrates the analysis of supporting of orthogonal data. So we generated here cage peaks of the same samples, and Scanty can process them and tell you how well these orthogonal data support your transcript models. But the same for the Illumina reads, so how well the Illumina reads are supporting the junctions, and the quant-seq peaks were indicating the terminations of the 3' ends of the transcript. So these orthogonal data, who well is supporting what the long reads methods provide. Okay, so I'm going to give you here now some examples of how we analyze the data, and I will try to explain this that you don't get lost. So here we have a comparison of the proportion of transcript detected as full splice match. These are the known, and the incomplete splice match, maybe fragments, right, by the different methods. And here is the percentage of novel in catalog versus novel not in catalog, right? So this is known, this is novel. We have here the names, right, of the different tools, and in colors we have the combinations of library prep and sequencing method. Okay, so what do we see here? So what you can see is that the names, right, of the tools come together, right? So here you have right? So one thing that we see is that the tool, the analysis tool, trying to give you the same result regardless of the data. And then tools like the ones here, like Bamboo or IsoQuant, they tend to give you a lot of full splice matches and very few incompletes, and also very low level of novels, right? And these are the tools that are heavily based on the reference. So somehow, if they are heavily based on the reference, they are giving you the reference. Then you have the other tools that were kind of in between, that have more like half of the transcripts are in the category of the known, and then you have a higher percentage of novelty, especially the novel not in and the novel in catalog, these new combinations, right? And then remember this tool that is not using the reference is kind of the one that gives you the highest number of novel not in catalog, so has the highest discovery power. However, you know, the known transcripts are much less in comparison. So in general, tools that provide a high number of full splice matches of the known transcripts are using the reference notation. We have this orthogonal support. So how well are these models supported by orthogonal data? So again, here we compare that are supported by the reference or by the cage or quant-seq data. So what we see again is that these tools here, right, the orange ones, are very well supported by the reference. However, you know, the support by cage data is around 60–70%, right? And even in the case of the 3' end support, they tend to be lower in comparison with the rest, right? However, the ones that have more discovery power, maybe they are not as well supported by the reference, but in general are better supported by the orthogonal data. And Lyric is somewhere here, well supported by the orthogonal data, but less by the reference. So tools that use the reference annotation are better supported by the reference annotation than actually sometimes by the orthogonal data. Okay. And also, the conclusion here is that a significant number of transcript models are supported by orthogonal data, but are not in the reference. So this is not directly extracted from this figure, but that was what we found, indicating that we have, you know, probably a lot of transcripts that are not in the reference yet, but are actually expressed in our samples. Okay. So this is like a general how they did with the scanty categories, but how do they compare? So what we are doing here is indicating the number of transcripts that were detected by an increasing number of pipelines. We had in total 47 pipelines, combinations of library preparation methods, sequencing platform, and analysis tool. So that were the number of data sets that we had to analyze for each of the samples. So here is one pipeline, and this is 47, and this is in the log scale, so the number of transcripts detected. So when you see that actually the highest in class in number were those that were detected only once by one combination of these three elements, right? And as we request that the transcript is detected by more, this is progressively decreasing. And actually, there were something like probably less than 10, around 10, 20 transcripts that were detected always by everybody, right? So that's kind of scary. And here, this is the scanty categories associated to this data. As you can imagine, those transcripts that were rarely detected by few pipelines were enriched and novel in catalog, novel not in catalog. So all these novels, but those that were consistently detected by many were mostly full displacement, right? So the consensus are in the known. Actually, if we say, okay, give me a kind of high confidence transcript model, so those that were found at least in 3 samples, and at least by 3 tools, those are basically, here are great majority are the full displacement. We have quite a number of novel in catalog, right? So they are these novel combinations, incomplete splice matches, and the novel not in catalog. And actually, of the other categories, we did not have many that were consistently detected. And if you ask, and which were the tools that were mostly giving these results, were Bamboo, Isoquant, those that are using the reference, were giving the best predictions of the reference, okay? So this is like the consensus is really in the known, but there are quite a novel of consistently found also novel transcripts here. Okay, so far what we have seen here is that we are just kind of analyzing the data, but we do not really have a ground truth. So to assess the ground truth, we have different ways. One was the usage of the SIRS. This is synthetic RNAs. And here is on these synthetic RNAs, which is the sensitivity and precision for the different tools with the different combination of samples, right? So what you can see is that in general, all the tools were very high in sensitivity and precision, right? Although they were giving kind of different results as well. But it was only Lyric and Talon that have of not as good values. Lyric was performing better, clearly better with PacBio than with Nanopore. Okay. However, we have to say that the SIRPs are known transcripts, so you have them annotated in the reference, so we can assess in this way the ability to detect novel transcripts. We can do this partially using the simulation data, the computationally simulated data. So we simulated data and we simulated also novel transcripts. And here you have the performance in a little different format, but we have here sensitivity and precision for the known, right? In the simulated data, sensitivity and prediction for the novel, right? So what you can see here is a different, and okay, red is PacBio simulation and blue is Nanopore simulation. I will talk about PacBio because the results with Nanopore are a little bit more lengthy to explain. But what we see here is that in general, as we expected, right, the known sensitivity and prediction are okay, but however, things are not going as well, both in sensitivity and prediction with the novel. So probably the one that is performing best in that sense is Mandalorian, but other tools, for example, Bamboo was giving great results on the known, but have like average. Flare was not doing bad. Spectra was doing a very high sensitivity, but very low precision. So, and the same with Lyric. So they have kind of different, they struggle a little bit more to get good results at the novelty. Other thing that we did was to use gene code experts to curate data. So they curated 50 different loci and they called transcripts directly from the reads, right? And then we assess the performance against this curated of manually annotated data. Again, here we have the different tools and different combinations of library preparation methods, and whether they are only using long reads or all combination with short reads. It's a little bit complex, but in general, we see that at the non-transcript level, we have, in general, good precision, right? And sensitivity is very, it's okay for most of the tools, but there are a couple of them, like, for example, Lyric. This one is Flames that is kind of struggling in finding those curated transcript models. But however, for the novel transcripts, they kind of really, everybody failed. most of them failed to find them. In some cases, they were a kind of around 50% sensitivity. Precision was good in some cases, but that was just because there were very few transcript models called. Okay. So sensitivity for novel transcripts is in general low. And what we did was, okay, let's go to the lab and let's try to verify in the lab with isoform-specific primers whether these novel transcripts are real, right? So what we did, we selected in total 100 transcripts that were in different categories. Some were full square matches, but we also have the novels. And we have novel transcripts that were consistently detected by many pipelines, and some others that were detected only by 1 or 2 or 3. Right? And we tried to amplify them. So what we found here is that for the known, for those that were consistently detected, whether they were already known or novel, we had a very high validation rate. Sorry, in the thick color is the validation rate. They were really, really validated. The ones that were rare, right? Both novel in catalog, novel not in catalog. I mean, the ones that were validated, even if they were found in 1 or 2 samples. Well, that was surprising. But even the novel not in catalog, it was like half of them were found, right? So it's like 50% is not good, not bad, right? We were surprised by these results. So many of the rare things were actually validated experimentally. And if we see, okay, so how many samples have to see a transcript to be actually validated? I mean, even, for one sample, we could still validate like most of them, like something like around 70% of the ones that were found only once in one sample, and if were found in more than one sample, then the validation rate was even higher. Okay, so we were supported by this high validation of reproducible novel transcript, but also like 50% validation rate in the novel not in catalog category. So the conclusions for the challenge 1 are here, like, well, I think that the long reads can help you to identify transcript with great precision, but results actually depend more on the algorithm than of the data, because some algorithms tend to give you the reference, and that is kind of biasing everything. So when you have a reference. Globally, I think that the combination of CDNA, PacBio, or R2C2 and Nanophore is what is giving you the best data to start with. And here was just a number of tools that are performing better than the others, right? For the reference annotation. Precision in general is better than sensitivity, especially for the novel. But the consensus in the discovery of novel transcript is very challenging. It's not that they are not there. So quite a number we could validate experimentally, but it's like they are not really consistently found in several samples. So maybe they are just found in few, but we can still find many of them. Okay, so that kind of poses a challenge. So which of the novels are the real ones to believe? Okay, so that was the conclusion of challenge 1. I don't know how much time do I have. We are okay. Okay. So I want to talk a little bit about challenge 3. Sorry, that was identification without the annotation, right? So you can already guess what the result was here, right? Here we use the mouse sample and the manatee sample. We also had the spikings. Only cDNA was evaluated, so only this library preparation, and again, the different technology. Right, so we asked a good transcript model, but without not using the reference annotation at all. But they could use the reference genome, okay, but not the annotation. For this challenge, we only had 4 participants. The tool Bamboo, IsoQuant, RNA Bloom, and RNA Space. That was a pity, but that's what we got. And here you have the number of total isoforms of transcript that were reported by each tool. You can see that there were very, very different numbers. So RNA space were kind of exploding in the number of isoforms that were detected. And here the structural categories, right? So you can see already that now it's not everything. It's not everything. Even Bamboo and IsoQuant is not many full splash matches that we had before, but also a lot of novel isomers, which are the rest of the colors. And even here, for example, with RNA spades, we have a lot of intergenic, right? So somehow they are capturing those that not the other were capturing. So very different results in this case. Two of these tools also gave us predictions when using the reference, so we could compare the same tools, right? What happens when it uses the reference annotation or does not use the reference annotation, and this is the result. And for the different type of data, and what you can see is that in general, right? So where they were blue, now they are kind of half, right? So when they have the reference, they can predict this very well, the mixed transcripts, but in absence of that indication, they are, for example, giving us a lot of incomplete splice matches, so we are not able to resolve these high prime ends of the transcript. So this is something that you saw a lot. Also, many more novel in catalog. So in general, you can see a big difference with the same tool, whether you are supported by the reference annotation or not. And well, we can see also some other characteristics, whether again the support by cage. This was only for the mouse data, by the way, of course, because the manatee we did not have that. But you can see here, like for example, in general, even if it was not perfect, Bamboo and the Shockpans Bamboo was giving kind of 60% support by case. Isoquant not as good, but the other two were even worse. And the support by Quantsec was around 60% for Bamboo and Isoquant and less for the others. We also look whether the junctions were canonical or not. Bamboo and Isoquant, fine, but the other two were giving a lot of junctions that were not canonical, so we were not even resolving that properly. And whether the junctions were supported by Illumina reads, Bamboo and Isoquant was giving us well-supported reads, and the others were around 50%, right? So clearly, even not perfect, these ones were kind of doing a better job. If we look at what happens with the SERPs in the absence of the annotation, as you can already expect, you are kind of having very different results. For example, with Bamboo and Isoquant, that is what we were better here. The prediction was kind of okay, but the sensitivity was really low, and here the prediction was kind of half, right? And I just want to give you the result as these two were given in chance one for the same data, almost perfect sensitivity and prediction with the reference annotation, but of course not when you don't have this support to guide you. So the conclusion for challenge 3 is that that's as you would expect. The accurate annotation without the reference is challenging, right? And it's hard to find this good balance between sensitivity and prediction. Tools like Bamboo and Isoquant, and we did not evaluate many because that were the number of submissions that we got, were in general good. Maybe poor sensitivity, but in general good support of transcription start and termination sites and the junctions. I think that one conclusion is that you have to be cautious when using these reads to annotate your genome if you really don't have a good annotation to help you. And probably it's not a bad idea to get some orthogonal data to kind of help you in supporting these transcript models. And these were the conclusions of challenge 3. So given that, we thought, okay, what can we do here, given this to try to help in the field of transcript annotation of new species using long-read methods. And we started to work a little bit on that. It's not that we have done a lot. It's not that we are actually experts in genome annotation, but I will show you a few slides of how far we came so far, right? So the data is how to best use this data for the normal transcript annotation, okay? So the idea was the following. This is where we started with, and that's that's the difficulty to define without the annotation. And the question was like, can we just combine ab initio methods, like a computational prediction, with long reads? And so which is the best way of doing that? right? So we try to evaluate whether it's better to use the reads alone or actually the transcript model, right? To support ab initio models with the long read state. So that was kind of bioinformatics master students' thesis. So this is the analysis. So what we try is like, okay, let's take the PacBio and the nanopore data, let's put it together also. And we can use the long reads, just the long reads to support. It's like the evidence-based ab initio prediction. We take the reads, create transcript models with ISO63, which is the PacBio official method, or FLAIR, which is something that can process both PacBio nanopore and the combination, create the transcript models, and then use this as an evidence to feed the ab initio methods, or just use it just like this. And this is a little bit the results of what we obtain. When we do that, here is again sensitivity and prediction using the PacBio nanopore of the combination of both. When we use the reads, the transcripts, and when we just at the genes, right, at the gene level. So what you can see, for example, here, right, is that when we use the transcripts, and the PacBio in general, so we will be having good sensitivity and prediction, right, with the transcript models, or when we are using the mix. The nanopore alone is kind of giving more trouble to find that. And this is kind of a different way of looking at that, right? So this is just the ab initio method, or with the evidence by the transcript. And what you can see is that sensitivity and prediction and precision, sorry, is the exon and the gene and the transcript level is kind of improved slightly when we use the evidence driven with the reads, with the transcript models. And also the identity when we use this evidence to support by transcript models is a little bit better than when we don't use that. We took that to annotate the manatee genome, in which we really had low quality data. I have not shown this in chapter 3, in challenge 3, but the data that we got from this field sample was not as good as the mouse. But then we did the same, right? So try to do the use just the reads, just the definition of with the transcript models. And what we can see is that when we provide the transcript models, in this case, the PacBio transcript models to the ab initio methods, we can get a higher number of single copy, well-defined postcode genes, right? So it seems that this kind of transcript model that is a kind of more consensus, clean, a little bit more robust data actually helps better than just using the reads, at least with the data that we use at that time. And this is one result of the manatee. We were trying to get a good, for example, a good annotation of the one of the subunits for hemoglobin that was really very badly annotated in the genome. That was the consensus in some other species, and then with our new data, that's we got like a more a gene prediction that was much better than what we had before. And with that, I conclude here. So the evidence annotation reduces false positive in the predictions, especially when you are using the transcript models, and PacBio in our case was better, and we could somehow improve the annotation of the manatee genome. And with that, I conclude. This is my current team, but this is work that we do with a lot of people that are not here. They are not in the lab anymore. Lorena was the creator of Scanty 3, together with Manuel, and other important person here to mention is Alejandro Paniagua, that is, where is Alejandro? Here, that did the last bit of the ab initio methods. And thank you for your attention.
Thank you very much, Karen. Very interesting talk.
Yeah, thank you very much. Very helpful work that, yeah, we all benefit from. So I have some questions and probably more than I can ask right now. One is, I may have missed it, but did you check for correlation between coverage or expression level and isoform or novel isoform identification, or just isoform number for a gene?
In our case, or with the tools that we need, okay, first disclaimer, right? Not every tool analyzes all the data. Oh, perdón.
Sí, no. Para que te ganen.
Perdón. Okay, so not every tool analyzes all the data sets. Okay? So that means that we could not really we did not have like a full experimental design because it was the decision of the submitters, right? So the developers of the tool, right? And so which data sets to analyze. So for example, Lyric, I think that, some tools were not analyzing nanopore data, for example, only big bio data, right? But we did not find necessarily that there were more transcripts, more good quality transcripts, right? Detected by more reads, right? It was more important that the quality of the read was good and the length of the read was good. We did not have more transcripts, for example, with direct RNA sequencing that was giving shorter reads. We did not. Any of the tools provided that, right? Some tools, they were kind of very promiscuous, right? And they will give you a lot of transcript models regardless the number of reads, right? It's probably within the same tool, right? If you have a little bit more of data, you will have a little bit more of reads, but that was not the major contributing factor to the result.
Why you would fail to identify an isoform then? If it's not a detection level issue, or is a complexity of gene, a number of exons, or there is anything telling you, generally speaking?
Were giving us a lot of reads. These reads were of lower quality. That was our problem, right? So we were having a lot of reads with the combination. A lot of reads were generated with nanopore, but the quality of the data was worse. So they were not able to give good transcript models in the end. The ones that were giving us the data was cDNA PacBio and R2C2 nanopore, but in this case we had less reads, right?
If I can, how would you compare the transcript model that you use for ab initio in the challenge number 3 between long read and short read? So if you had to do a de novo assembly of the...
Yeah, that.
Okay. Because that's what we do in my lab. We do, because we started before, but then we use ISO-seq to improve at the very end. We use the ISO-seq libraries to put on top the de novo, ab initio done on.
This is still pending. I know that we have to add that. Yeah. Thank you.
Nobody else?
Thanks for a very nice talk. And I have a few questions. And the first one is, I also saw that there is a direct RNA sequencing. Do you check also for like other information related to methylation, maybe? Okay, yeah, there was just, yeah, I guess it was out of context. I was just curious. And the other one is if you checked also for expression level.
Yes.
And if they are like, so if in your opinion, it would be feasible to consider like longer reads sequencing for quantitative transcriptomics.
Well, this data is now, so the platforms are giving you much higher throughput, right? So that's what happened. It's a challenge too. I did not go into that because I did not have the time, but you can find the paper on the bioarchive and hopefully also publish very soon. So the major conclusion was that nanopore was better than PacBio for quantification on the reference annotation because of course you need to have a good transcriptome first, right? So, but if you forget about that, if you kind of only focus on the transcripts that are annotated and do not even try to find new ones, right? And then you use the reads just to find those. We were getting better results with the nanopore with these 20 million reads than with PacBio that were producing 2 million. Now we have samples with the new MASIC and review protocol in which we have 50 million reads with little effort, and that's completely changing the conclusion of this. You know, for the non-transcripts, right? At least what we found is that it was not really, it was good, right? It was, the quantification was good, but was comparable to do it with short reads on, you know, the non-transcript. Now, so my guess is that when we kind of fix the problem of the novels, that we see that there are many, right? And this is something that we can fix with the long reads and with the new quantification, with the new throughput, then you will have like the best of, right? So the ability to detect novel transcripts and the ability to quantify them properly. And that this is kind of here, I guess. Right.
Any other question?
Only to expand the question that just Marco did. Do you think that these methods will help us in the challenging about the haplotype specific expression analysis and so on? Because one of the struggle that I encountered in the short reads.
I think so, and people are using that already for resolving haplotypes, at least for diploids. I think that also more complex genomes. PacBio have also some tools already doing that. We are now starting a project in which we are also evaluating that specifically for plant genomes or plant transcriptome. But yeah, in principle, yes, because you know you have activity and then you can have the variations. So but then you don't know. Yeah, you could also probably do that. But if you have if your genome is not exactly the reference, you know, that's the problem.
Any other question? You can see that we need to start worrying now about this rare. If you were able to later on, maybe that.
Yeah. So I think that we have done some analysis trying to quantify this fraction of rare transcripts. Of course, I have not done this in plants. I have done this in mammalians, in mouse, and in human. And that's kind of probably around the 20% of the transcriptome is in this gray area. And I think the challenge now is to distinguish that from the technical errors that are out there and designing the strategy.
but we have evidence that yeah, that you have around these levels of funny transcripts as well.
So thank you once again.
Thank you.
It's a wrap for today. We'll see each other tomorrow for more interesting talk and enjoy the